So we are sharing from our end.
Okay.
Good morning, everyone. Welcome to the 6th day of MMTT 2021 International Workshop. Today, the first talk will be given by Dr. Manavalan Balachandran. Dr. Manavalan Balachandran received his PhD in 2011 from Ajo University, South Korea. After completing his PhD, he moved to Korea Institute for Advanced Studies, Seoul, for his postdoctoral research. After completing 2 years postdoctoral research, he moved to Department of Physiology, Ajo University, as a research professor, and currently he is an assistant professor in the Department of Physiology in Republic of Korea. He is also an associate member of CHIAS, Republic of Korea. His main research interests include prediction of protein structure, machine learning, data mining, computational biology, and functional genomics. He has developed more than 25 bioinformatics tools and programs and scripts and software, and they are all available on his research papers and on his servers. They have been widely accessed by research community around the world. He has been listed as top 2% scientist in the world in bioinformatics area. And he is the only person in this category from whole South Korea. He has published more than 40 research papers. They are highly cited, and he is very successful in doing protein modeling, protein structure prediction, protein folding, and several other machine learning and bioinformatics tools. Welcome, Dr. Manavalan Balachandran. Thanks for coming. Now you can start the talk.
Okay. First of all, I would like to take this opportunity to thank organizers, especially Professor Hemant, for giving me an opportunity to present my research work in this wonderful international workshop. I am Balachandran Manavalan, working as an assistant professor in the Department of Physiology, Ajo University, School of Medicine, Suwon, South Korea. Today, I am going to talk about development of therapeutic peptides using machine learning. Next slide. So this is the contents of my talk. So first, I will talk about peptide-based therapeutics, followed by a few prediction methods like anti-cancer peptide prediction methods, hemolytic peptide prediction methods, anti-hypertensive peptide prediction methods, followed by critical evaluation of existing peptide therapeutic prediction methods, followed by classical approaches that we have introduced recently. And in the summary, I will give you some key to successful model development. Next. So this is the first peptide-based therapeutics. So peptides represent a unique class of pharmaceutical compounds, molecularly poised between small molecules and proteins. So yet biochemically and therapeutically, which is different from small molecules and proteins. Peptides are naturally occurring biologics with the size ranging from 2 to 50 amino acid residues in length that play a crucial role in as anti-infectives, growth factors, hormones, biological messengers, and neurotransmitters. The discovery of peptide hormones, including insulin, vasopressin, oxytocin, and gonadotropin releasing hormone, spurred a major advancement in biology, chemistry, pharmacology, and other state-of-art technologies in modern drug discovery. In 1921, the breakthrough of insulin, the discovery of insulin, which was considered as one of the monumental scientific accomplishments, which later became the first commercially available peptide drug, paving the way for the identification and the screening of other therapeutic peptides and discovery of peptide-based therapeutics. So certain peptides have been identified to possess a wide range of biological properties, including anti-angiogenic peptides, antibacterial peptides, anti-cancer peptides, antifungal peptides, and so on. Because of their high specificity, selectivity, efficacy, reduced toxicity, and short half-life, peptides are relatively safer than the traditional drugs. Next. Peptide drug market. So peptide-based therapeutics have been experiencing revival in global market with the collective sale of more than 20 billion US dollars in 2017 alone. So in 2024, actually the sale of peptide-based therapeutics are expected to surpass 15 billion US dollars. So you can see the figure on the left-hand side. The number of peptide entering into the clinical trial, that's the orange color graph, which is slowly increasing. Okay, so on the cumulative peptide approval, the gray color, so which is also like a steadily increasing. So this clearly shows that the last 2 decades, the number of peptides entering into the clinical trial is steadily increasing. The figure on the right hand side, because I cannot use pointer, so just I'm mentioning the figure. So figure on the right hand side, you can see here the number of peptides entering into the clinical trials, especially in the last 2 decades in 2000 to 2009 and 2010 to present. So it's also like steadily increasing. The orange color represents the peptide, the therapeutic peptide with the length of 2 to 10 amino acid residues. And this gray color is the amino acid length, which is 11 to 20 amino acid residues. Yellow color represents like 21 to 30, so on. So as you can see here, the peptide entering into the clinical trial is like a more majority of them less than 40 amino acid residues. Of those, like majority, this orange color, you can see that in the last 2 decades, like in 2000 to 2009, it's like a 70 peptides, which is residues 2 to 10 amino acid residue. So majority of them are 2 to 10 amino acid residue. So when you are designing a peptide, like keep it in your mind, like you can design a peptide which is much shorter than the longer peptide. So currently more than 100 approved peptide drugs are commercially available for patients with various diseases, and several additional peptide agents are in preclinical and late clinical trials. Next. So machine learning as a complementary to traditional approach. So identifying suitable peptides. So how the experimentalists identify the peptide therapeutics. So you can see here on the left-hand side, there is a large number of peptide sequences in the peptidome. Okay, so what they do, they randomly select some group of peptides, and then they can study their activity and then whether it has some their desired activity or not. Otherwise, what they will do, they will take, you can see the picture on the right hand side. They will take the entire protein sequences and then cut into fragments. Generally, they use some kind of like overlapping window of 1 or 2 residues, and then they test the activity of each segments. So just imagine, like if they do these kind of approaches, like traditional approaches, it will take a lot of time, and also it's highly expensive. Okay, so overcoming these limitations requires novel and efficient computational approaches that can be used to uncover candidate peptides. Such approaches can be used to rapidly screen prospective peptides before the synthesis, thereby expediting peptide drug discovery. Machine learning based computational approaches can serve as a rapid and inexpensive pre-screening tools to efficiently navigate the vast combinatorial sequence space. So actually these will streamline the burdensome process of peptide drug discovery. Next slide. Next slide. Here I have shown why a sequence-based method is much preferred than the structure-based method. In sequence-based method, it is very easy to generate peptide sequences. Like you can generate millions of sequences with some computational programming language. So it's very hard to generate peptide structures. Sequence-based method can be applicable to a large-scale analysis, whereas the structure-based method cannot be applicable to large-scale analysis due to difficulty in 3D structure determination. From experimentally verified sequences, one can generate a huge number of, one can get a large peptide sequences data set. Once you have the data sets, it's like it will be helpful to develop a more reliable model. However, in docking studies, it will be carried out an interaction between peptide and then protein, and you can see the just an interaction. That's it. And machine learning based model generate a confidence score between 0 to 1. Like, for example, if it is anti-cancer peptide, if it is greater than 0.5, you can say like it has an anti-cancer activity. If it is greater than 0.95 or yeah, greater than 0.95, you can select that putative peptide with a high confidence, isn't it? So therefore, it is very easy to pick the putative candidates. Whereas in docking score, there is no well-defined range. Therefore, it's very hard to pick the putative candidates. And it's very easy to study the mechanism of action. For example, you can screen the peptide, millions of the peptide in a day or 2 days using machine learning based method and identify the putative peptides. So these putative peptides can be subsequently utilized for experimental studies. Whereas in structure-based method, it may give a glimpse of protein peptide docking interaction, but very difficult to study mechanism. We can use a structure-based method. So when we can use the structure-based method, so actually once you have identified the peptide with high activity from the experiment, and you can study the structure-based mechanism, like how these peptide binds with the specific target. So that in that case, you can use a structure-based method. Next slide. Here I have shown an overview of peptide drug discovery. So you can see here the databases. So databases is nothing but like peptide sequences with different functions that have been already reported. So those information has been collected and constructed primary databases. So utilizing those databases, so one can apply artificial intelligence as a machine learning based method to develop a prediction model, and that will predict the peptide therapeutic functions. So once you identify the peptide therapeutic functions, so those peptides can be chemically synthesized and in vitro studies, followed by primary assays, high throughput in vivo studies, followed by secondary assay, counter screen, bioavailability, etc. Actually, the selected peptide candidate, which has now passed all preclinical tests successfully, is given to human patients in a clinical trial. Next slide. Here I have shown general and specific databases currently available for the prediction of bioactive peptides. The first column represents the bioactive peptide, and the second column represents a database. Third column represents the description of the database. Fourth column is the year that database was developed, and the fifth column is the database link. As you can see here, the first column, this is a general type, and here we have mentioned only the 6 different peptide therapeutic function, like anti-cancer peptide, cell-penetrating peptide, quorum sensing peptide, anti-hypertensive peptide, anti-tubercular peptide, and anti-inflammatory peptide. So as you can see here, next slide, and next. Yeah, as you can see here, the lines, red lines. These are all the databases developed by Professor Raghava from IIT Delhi. So actually his contribution is significant in constructing primary databases for several peptide therapeutics. So generally collecting peptides from the literature search is a laborious and extremely difficult. So it can be achieved only with large human resources. So in this regard, Professor Raghava contribution to the peptide research is significant. Next slide. Next. So here I have shown like overall framework that for like 4 major steps involved in development of a reliable peptide-based therapeutic prediction model. So first is that data set preparation. So you have to construct the data set. So based on the publicly available databases, or primary literature search, and then you have to separate the data set according to the peptide function. Here I have shown 6 different functions. ACP is nothing but anti-cancer peptide. AHTP is the anti-hypertensive peptide. CPP is the cell-penetrating peptide. AAP is that anti-inflammatory peptides. ATPP is that anti-tubercular and quorum sensing peptide. So you can see here, next step is that redundancy reduction using CD heat. So it all the CD heat cutoff is all depends on the size of your original data set. If your original data set has a huge number, then you can go for CD heat with greater than 80% sequence identity, which means that the sequence that share a greater than sequence identity will be excluded from your data set. From there, you can select 80% of the data for the model development. That's a training data set, and the remaining 20% is considered as an independent data set to check the model transferability. Second step is that feature encoding. Here I'm not going to point out each feature, but like several features can be grouped according to the some function like composition based, position specific based, motif based, physiochemical property based, and evolutionary based. So each property has several feature encodings, and it has their own dimension. The peptide with varied length that can be converted into fixed length of feature vector based on these feature encodings. So generally these feature encodings contain some like irrelevant information and also some unnecessary information. So this has to be eliminated to improve the performance and also to increase the training speed. For example, if your data size is around 10,000 and then feature dimension is 3,000, it will take a long time to compute the develop the prediction model. So like if you do the feature optimization techniques and reduce the feature, let's say like from 3,000 you reduce to like 300 or 500. So the it will increase the training speed. And then you have to identify the optimal features. So once you identify the optimal features, it is always recommended to explore different machine learning classifier on the same data set, because these performances of machine learning is a data set specific based on my experience. So therefore, it is highly recommended to use multiple classifier on the same data set, like a support vector machine, artificial neural network, and deep learning. Decision tree based classifiers like random forest, extremely randomized tree, gradient boosting, and extreme gradient boosting, so on. This is the nearest neighbor is a very simple algorithm. And fourth step is that model training and selection. So you have to optimize each machine learning parameters using cross-validation technique. So depends on the data size, you can pick the cross-validation, either it's a 5-fold cross-validation or a 10-fold cross-validation, and you can develop the prediction model. And you can see the performance of all the model. If a model is statistically better than other model, and you can pick that as the final model. And you can see like if there is no significant model and you are observing like some group of model achieving like more or less similar performance, let's say like 82 to 84% accuracy, you can use all those model to construct an ensemble model, and that will be considered as the final model. So once you develop the final model, and you have to, the most important thing is that you have to check the model transferability, whether it is using independent data set, whether the final model is able to replicate the training performance on the independent data set. Next slide. So now I'm going to talk about anti-cancer peptide. Next slide. Here I have shown mechanism of action of anti-cancer peptide. Anti-cancer peptides are part of antimicrobial peptide that exhibits anti-cancer activity. Generally, these are like 5 to 50 amino acid residue in length and cationic in nature. Mostly they possess alpha helix as the secondary structure. Sometimes it folds into beta sheet, and sometimes it has an extended linear structure. As you can see here, the cancer cell. Cancer cell possess larger surface area due to presence of high number of microvilli negatively charged cell membrane, and higher fluidity of membrane, etc. These features allow cationic ACPs to interact with negatively charged membrane via electrostatic interaction. And once it binds with that cell membrane, it enters into in the first one, it enters into the mitochondrial membrane and it destructs the mitochondrial membrane. That's the ACP inducing apoptosis of cancer cell. Second one is that it binds with the cell membrane and it lyses the cancer cell. That is ACP inducing necrosis of cancer cell. In the third process, it inhibits the angiogenesis in cancer cell by ACP. In the final one, like it may recruit sometimes it may recruit other proteins, and then it inhibit and activate some process. Therefore, thereby it can kill the cancer cells. So these are all the mechanism of action of anti-cancer peptides. Next slide. So we have developed two anti-cancer peptide prediction method. First one is MLACP and second one is MACP-PRED. So MLACP was developed in 2017 and MACP-PRED was developed in 2019. So first I will talk about MLACP because that was my first peptide-based prediction method. So here I use the training data set of 187 ACPs. And 398 non-ACPs. So all ACPs are experimentally characterized one. Since there is no experimentally characterized non-ACPs, so generally random peptides are peptide with non-secretory activity has been considered as non-ACPs. So here also we have used the same approaches. In the independent data set, I generated two independent data set based on the existing data set, and also we also searched with different databases, and then we found some peptides. And this is the independent data set is two different data set. In the first step is that we extracted two different features like composition based and property based. So generally, it is essential to see each property you are calculating for the positive and negative data set, and there will be some differences between the positive and negative. That's a positive means anti-cancer peptide, and negative means it's a anti-cancer peptide. So these kind of variation has been inputted to the machine learning method. This machine learning can identify the hidden pattern between the positive and negative samples, and thereby it can capture those information, and then it will give a much higher accuracy. So here for composition based, I have used three different composition, and for property based, I have used 11 different physical chemical property. So all these combination feature combination we have generated. On the right hand side, you can see like overview of MLACP. We use the training data set, and then we optimize the machine learning parameter using both tenfold cross validation and the leave one not cross validation. Basically, we have used two different classifier, random forest and support vector machine, and we identified the optimal feature set, and then we evaluated using the independent data set, and then based on the optimal model, we developed the web server. Next. Here I have shown the performance of RF and SVM on the training data set. The first column is that features, second column is the Matthew correlation coefficient, and the third column is the accuracy, so on. As you can see here, the combination of four different composition, which gives the identical performance on the training data set. You can see the MCC is like 0.697 and 0.698, and accuracy is identical, 87.2%. So therefore, we selected this as the final model. Next slide. Next slide. Yeah. So we evaluated using the two different independent data set. You can see here in the case of HC data set, RFACP achieved like 94.6%, and then in the independent evaluation using a lead data set, it achieved 81.1%. So we have in our web server, it has both RFACP and SVM-ACP so that the user can make a consensus prediction between these two prediction models. So actually, these web server heavily accessed by the scientific community and several personal communication from a different experimental group. And as a result, we also noted that there are several disadvantages in the MLACP method. So actually, this was our first experience. So we observed several disadvantages. We didn't see these kind of disadvantages while developing the prediction model. So first is that imbalanced data set used to train the prediction model. So non-ACP is approximately 3 times higher than the ACPs. So generally, whenever you are dealing with the imbalanced data set, then there should be some special care should be taken while developing the prediction model. But we did not take any special care. As a result, you can see the independent data set using Lee data set. You can see lower sensitivity and higher specificity because our prediction biased towards the larger negative data set. So that's a disadvantage of the MLACP method. Next slide. Next slide. Yeah. So we addressed all the shortcomings of MLACP and developed a new method called MACP-PRED. You can say it's an updated version of MLACP. So we constructed the data set of ACPs and non-ACP, and we applied a threshold of CD hit threshold of 0.8%. So actually, this is a first instance where CD hit of 0.8% was applied in the peptide-based therapeutic prediction method. So we applied very much a higher CD hit cutoff, and then from there we generated a benchmarking data set for the model development, and then independent data set. So I will talk about how we constructed the data set in my latest slides. In MLACP, we used only 4 encodings. So here we used 6 different encodings. You can see here the amino acid composition, dipeptide composition, CTF, QSA, quasi-sequence order, and then binary profile. In physicochemical properties, we have used 2 different physicochemical properties like composition, transition, distribution, and amino acid index. And next is that we have optimized in MLACP, we did not use any feature optimization. Here we have used feature optimization like F-score plus sequential forward search. So these I will explain in my later slides. So once we find the optimized features, and the third layer, that's the 2-layer model constructions. So these optimal features inputted to the support vector machine separately and develop their corresponding model. And these predicted probability score of each model, that's the ACP predicted probability score. These scores are combined and again inputted to the support vector machine for the final prediction model development. In the performance assessment, we have used a 10-fold cross validation, and then model assessment based on the independent data set, and then we develop the web server. Next slide. Next slide. Yeah. So actually, this is the relationship among various data set related to the MACP-PRED data set. So this is not my slide. Like actually, I have taken this slide from the briefings in bioinformatics published recently. So they have used our MACP data set and named it as a Bhupati 2019 data set. So here I will show like how we have constructed the data set, because they made it in a very they made it neatly, and then it's very clear. So you can see here the first data set reported by the Tyagi 2013. This is Professor Raghav group, and they have constructed the training and independent data set based on the 3 major database. That's APD, CAMP, and DADB. In 2014, Hajishvi from Iran, they have developed the anti-cancer peptide prediction method using this APD database. In 2015, Vijay Kumar from Pondicherry University. So this guy, he constructed the data set based on these three database. Like primarily they focused on only on these three database. After that, cancer PPD published by Professor Raghav group. So it became a cancer peptide database. So using this database, Chen 2016, they constructed a training and independent data set. And then subsequently using the existing data set, we et al. In 2018, they have constructed training and independent data set. But in our case, in what we did, in MACP PRED, that's about by the 2018, we considered all the existing data set and consider as the only as a training data set. But independent data set we constructed based on the newly available databases like the updated version of DADB and new databases like DB, ASP, RAMP, and LAMP. So we consider the anti-cancer peptide from these databases only for the independent evaluation. So this way, like we have constructed the data set. Next slide. Yeah. So the figure on top right, top left. So we have evaluated the 7 different encodings using support vector machine, and then we assessed each encoding performance. So as you can see here, the blue and yellow, these performance is better than the other feature encodings, but we considered all the encodings, and then however we consider all the encodings. Next we compare the support vector machine with other classifiers like random forest, logistic regression, and KNN algorithm. On average, support vector machine performance is better than the other 3 classifiers. Next, we applied sequential forward search. So here I have mentioned the procedure for sequential forward search. So you can see here, the first step is that this is the original F1 to Fn. So that's the original, please point out. So F1 to Fn is the original feature dimension. So based on some scoring function, here I have used F score. So you can use RF score, that's a random forest based score, or whatever, like several scoring function is available. So you can use those scoring function and you can rank the feature in a descending order. And next step is that you can select like one feature at a time. And here I have shown like F2. So the first step is that to take one feature. And the second step is the top 2, top 3, top 4, like so on. And then each feature set inputted to the some classifier. Here I have shown XGBoost. So you can use any classifier and then you can train the model and then you can see the performance. And like once you feel like if it is an imbalanced data set, you can select like MCC as the parameter to select the best model. If it is a balanced data set, you can pick accuracy as the parameter to select the best model. The best performance is considered as an optimal model, and whose corresponding optimal feature set is considered as an optimal feature set. So this way, like you can carry out the sequential forward search. So basically, this is a computationally expensive procedure, but this is a more systematic way to select the optimal features. And next, we have shown the performance of 7 different encodings with respect to different feature sets. And you can see here the optimal performance we have shown, we have highlighted in the number, like the first value represents the number of optimal feature set, and and then next second value represents the accuracy. As you can see here, the optimal feature set, like although the feature dimension is in case of dipeptide composition is 400, we identify the optimal feature set is less, like a more on all the cases is less than 150. Next slide. Yeah, so next we compare the control and then optimal features. As you can see here, in the case of amino acid composition and amino acid index and quasi-sequence order, there is no much difference between the optimal features and control. Whereas in the case of dipeptide composition and binary profile and CTF, there is a small improvement, but in the case of the feature dimension is significantly reduced. Next slide. So black color is that MACP performance on training data set. And then you can see the baseline classifiers like other 7 encodings, the integrating all those information, it improves the performance significantly. You can see in the case of MCC, you can see how much improvement, it's approximately 2%. And next is that we have checked whether whether any hybrid features like what we have employed in MACP PRED, it can improve the prediction performance. So what we did, we can linearly integrate 2 features, 3 features, 4 features, so on, that is named as H1 to H6. So you can see here, again, the MACP PRED performance is much better than the hybrid features. And next, we evaluated our method using independent data set. You can see here, MACP PRED achieved like 91.4% accuracy. So when compared to our previous method like SVM ACP and RF ACP, our improvement is significant. You can see the using a p-value cutoff less than 0.05, it significantly outperformed all 3 methods. So here also like our drawback of this SVM ACP and RF ACP is reflecting. You can see like lower sensitivity and higher specificity. Okay, so here we have shown ROC curve like MACP PRED is significantly better than the other prediction models. Next slide. So this is a large-scale comparative review and assessment of computational method for anti-cancer peptide identification. So this published last month from Jiang Yingsang group from Monash University. So he also used our data set for the model development. Next slide. What he did, he used our data set. And then he employed 5 different encodings like amino acid composition, PAC, and composition transition distribution, quasi-sequence order, CKS, AAP, etc. So basically, they have evaluated like 25 different encodings and then selected only the top 5 feature encodings. And then all these feature encodings are combined, linearly combined, and then they identified like they got like 2,289 features. And then they applied feature selection techniques. So their feature selection technique is slightly different from our standard approach. And they inputted to the 3 different, they developed 3 support vector machine based model and one light GBM and one KNN based model. And all these model predicted probability, again, inputted to the logistic regression, and then they developed the final prediction model. Next slide. Yeah. So this is a performance comparison of different machine learning classifier on 10-fold cross-validation test using benchmarking training data set. So here the first 7 like a KNN2 SVM C, it's there like baseline models. All these model integrated to the ACP PRED stackle, that's their model. And you can see here, like in terms of MCC, integrating all these model, the MCC is improved from 81.9 to 83.9. And then they have compared with the MACP PRED. You can see the last 2 methods, ACP PRED, stackle and MACP PRED. Can you see any big difference? So it's like the performance is the same as MACP PRED method. Next slide. Above one is that cross-validation performance, which I discussed in my previous slide. So there is no much difference between the red and blue on the 10-fold cross-validation. Whereas the independent evaluation, you can see here the difference between MACP PRED and ACP PRED stackle. It's like minor improvement. You can say like a 1.7% improvement in terms of accuracy and in terms of Matthew correlation coefficient, and then improvement is very minimal, like 1% improvement in the accuracy. So we got like a 91.4% accuracy, and they got like a 92.4% accuracy. So this clearly shows that our method MACP PRED performance is robust, and already it's an optimal one, and it cannot be further significant. It cannot be improved significantly further by different approaches. And this is the case study they have shown. So actually they have collected these 4 peptides like a helical 1, amphiarc 1, amphiarc 2 and gradient 2 from the recent study. So these are all the 4 peptide sequences. And third column is that prediction. So among those 4 peptide, like gradient 2 is active against breast adenocarcinoma cells. The remaining 3, amphiarc 1, amphiarc 2, helical, are active both against MCF7 and also lung adenocarcinoma cell lines. So among those 4 peptides, their method, that's ACP pred stackle, it predicts just only one peptide as an ACP. But so I wanted to see like how my this is the like you can show your method whether it has a practical applicability or not. So therefore I collected these peptide and then inputted to the MACP pred method, and then I have shown the result here. You can see here and below the results. All these 4 peptides have been predicted as an anti-cancer peptide by our method. And most importantly, these peptide is not present in training data set of existing methods like even Jiang Ying Sang method and also my method. So these peptides is not present in our training data set. And also our prediction, all 4 peptides predicted as an anti-cancer peptide. You can see the probability value. It's like more than 92%. So this clearly shows that practical applicability of our MACP pred method, even when compared to the newly proposed method. Next slide. Next slide. So I'm going to talk about hemolytic peptide. What is hemolytic peptide? The term hemolysis indicates the immature destruction of red blood cells before reaching their expected lifespan of 120 days. The consequence of such action could be seen in the reduction of hemoglobin levels in the blood, which further exacerbates and its decreased oxygen carrying capacity. So here I have shown mechanism of action of hemolytic peptide. So these RBCs is binds with this peptide binds with RBC, and it undergoes hemolysis. It will destroy the red blood cells. So far, like a number of potential therapeutic peptides falling at a clinical trial could be attributed to the toxicity profile, particularly the undesirable toxicity arising through the hemolytic activity towards a eukaryotic cell. So therefore, like hemolytic peptide is an undesirable property for constructing therapeutic peptides, and timely identification of such hemolytic peptide is deemed to be an essential step in the development of peptide-based drugs. So therefore, in this study, we proposed a computational model that can discriminate HLPs from the non-HLPs. Next slide. Next slide. Okay. So here we have shown the flowchart of HLP pred fuse. So there are 2 data sets. The first one is that like whether the peptide is a HLP or non-HLP. Second one is that like a high and then low activity. So we used both the data set, and then we developed a prediction model. So here we employed like 9 different encodings, and few are like composition based, few are position specific based, and then few are physicochemical property based. And then here we have employed 6 different classifiers: random forest, extremely randomized tree, support vector machine, gradient boosting, ADA boost, and KNN algorithm. So basically we carried out a 10-fold cross validation and then developed the corresponding model. So for each classifier we got 9 prediction model, and total we got 54 prediction model. So all these models predicted probability has been concatenated and then again inputted to the extremely randomized tree. Here also in we did not randomly select this extremely randomized trees. Like we also explored other classifiers and found that ERT is more suitable. Therefore, we employed ERT for the final model construction. So based on the final model, we proposed a two-layer framework. For a given input query, like the first layer model will classify whether a given peptide is a HLP or non-HLP. In the second model, it will classify whether it's a high hemolytic activity or low hemolytic activity. So these kind of information will be helpful to screen the peptide with the low hemolytic activity. Next slide. This is the first layer prediction performance based on the like whether the peptide is a hemolytic activity or non-hemolytic activity. So we can we assess the 6 different classifiers on 9 different encodings. You can see here in terms of some encodings like a amino acid composition and then AAI, there is no difference in the performance. There is no performance difference in the classifier. Whereas in the case of CTD, CTF and BPF and GTPC, we can see some kind of performances differences between the classifiers. So we combined all these classifiers predicted probability value, and then we integrated and then we developed the HLP red fuse. In the figure B, you can see here the red color. It's like the improvement is not like a significant, but it's very marginal. And when compared to the best like a best model from the figure A, like yellow and green, so the red is like a slight improvement. Whereas when we compared our HLP pred with the existing method, that a hemo-PI and a hemo-pred, so hemo-pred, so still the improvement is like approximately 3% is higher than the existing method. You can see in the figure C. Next slide. So here we employed the same procedure for whether activity prediction it's a high or low. So you can see here the improvement in terms of MCC. You can see the figure B. The improvement is like a much higher than the first layer prediction. And when compared to the existing method, like in figure C, like the HLP pred fuse improvement is significant. Next slide. So here we have evaluated all the baseline models and also HLP pred fuse. The figure A is the first layer and figure B is the second layer. You can see the gap in the MCC between the baseline model and then HLP pred fuse. In the case of training, it's the gap between the best baseline model and then HLP pred fuse was very small, but here the gap is increased. This clearly shows that HLP pred fuse has is more robust than the baseline classifier. In the case of second layer prediction, activity prediction, the performance is like is slightly better than the best baseline model, but in the training data set it was opposite, like it was like significantly better than the baseline model. Here the gap is like very small. Next slide. In independent evaluation, we evaluated HLP pred fuse with the existing method. Hemo-PI, this method is from Professor Raghav group, and this is a hemo-pred. This is from Thailand group. So we evaluated using the our own independent data set. So you can see here the performance of HLP pred is significantly better than the existing method in terms of p-value, the last column. Whereas in the case of activity prediction, so our performance is similar to the hemo-PI method. So the improvement is like just 1%. So we saw next slide. So here we have shown why HLP pred fuse is doing better than the baseline classifier. As you can see here in the first layer, the dot red represents the pink represents the HLP and then blue represent non-HLP. The original feature dimension of AAC, AAI, CTD, DPC and QSO, there is a separation between positive and negative is there, but still some data points are overlap. But through the feature fusion approach, you can see the figure F of the first layer. You can see the two clear separation between the positive and negative sample. Very few sample overlap with the overlap in the opposite side. So that's the result. That's the reason like HLP PRED is doing much better in the first layer. Whereas in the second layer, you can see here, like all feature encodings that overlap, but our feature fusion approach, there is some separations there, but most of the data, like some data points is overlapping between the positive and negative samples. As a result, in the second layer, the performance is like approximately like 80%. So therefore, in the second layer, we need some kind of novel feature encodings that can highly discriminate between positive and negative sample is needed. So this work was published in Bioinformatics in 2000 last year. Next. So I'm going to talk about antihypertensive peptide. Here I have shown a mechanism of antihypertensive peptide. This is a simplified scheme of blood pressure regulation showing the relationship between the main mechanisms. So here there are three different mechanisms: anti-angio, angiotensinogen, nitric oxide, endothelial system. Here the enzymes are shown in the red color, and the potential targets of peptide that mediate anti-hypertensive effect is shown in yellow color. Next slide. So here the same strategy, like we have constructed data set based on the existing database and also like our own literature search. And then we extracted a feature like in this study we have extracted a 51 feature descriptors. Actually, this is the first time a large number of feature descriptors employed in peptide-based therapeutic prediction method. And also this is first meta predictor for peptide-based therapeutic function prediction. And all these feature inputted to the extremely randomized algorithm, and then we developed the prediction model. So all these prediction model of models predicted probability value were considered as an feature vector. Next step is that we rank the features using random forest. So in my previous slide, I have shown I have used F-score for ranking the features. Here I have used random forest, and then all these feature inputted to the 4 different algorithms like a random forest, support vector machine, gradient boosting, extremely randomized tree, and then carry out the sequential forward search and to identify the optimal model. And the final model predicted probability score, again it will be integrated in terms of like we will take the average of the predicted probability score to compute to classify whether it's a antihypertensive peptide or non-antihypertensive peptides. Next slide. Yeah. So here in figure A, we have shown the ranking of the features. And then figure B, we show the sequential forward search, and the optimal performance is shown in the bracket. That's the feature dimension followed by the accuracy. In the figure C, we have compared with the control. When you compare with the control, the improvement is very marginal. You can see that only the green color. So RF bold is the control, and then without bold is then optimal feature based performance. The improvement is very marginal, and then we integrated this score, and then we developed the like a final model. You can see here the MMTT PRED. The performance is like similar to the other classifiers like support vector machine, extremely randomized tree, etc. Next slide. But when we compared with the MMTT PRED with the baseline predictors, the improvement is significant. You can see here the purple color of the MCC and the ACC. When compared to the baseline classifiers, this is like 51 feature descriptors, so 51 baseline models. So these MMTT PRED performance was significant. And we compared the existing methods on the same benchmarking data set. So we have, you can see here like PAP and these two methods. And you can see the improvement is significant, like approximately 6% improvement in our method. So the improvement is mainly because of the approach that we have employed in this method. Next. And then next we compared the MHT performance and then existing method performance based on the independent data set. You can see here the first table, the last column, the p-value. It's based on the AUC. So MHT performance is significantly outperform the existing methods. So actually why MHT is doing better, because the same the approach we employed that can distinguish between the positive and negative samples. As a result, the performance is better than the baseline classifiers. Next. Yeah. So here I have shown apart from these three methods, we have also developed several peptide-based therapeutic prediction methods. So all these peptide prediction methods have been heavily accessed by the research community. So which is evident from the citations. So if you are interested in developing therapeutic peptides, please use these methods. Next slide. Yeah. So generally, these are all some worldwide competitions. In the worldwide competitions, CAPRI's for protein-protein docking and for structure prediction, and CASP is specifically for protein structure prediction. Sample challenge is statistical assessment of the modeling of protein and ligands, and the CAMEO is that continuous automated mode evaluation and dream challenges. So I think okay, I'm telling these things because those who are not aware of these competitions. So in this competition, what they will do, they will give you the problem, okay, on a daily basis. On average, it's a daily basis or once in 2 days for 3 months or 4 months. So they will give you the problem. So using your algorithm, so you have to solve the problem and then you have to submit the solution. Okay, so once this process will continue for 3 months or 4 months. So once you submit the solution, they will compare the solution with your predicted solution with the actual solution and see how much you are deviated from the actual solution. So based on that, they will rank the method and then they will tell who is the best predictor and so on. So specifically, I will tell about the CASP. CASP is it's like a protein structure prediction. So they will give you the sequence. Okay, the problem is the sequence, and the solution is that you have to submit the structure. So actually that structure will not be publicly available. So you have to use your a developed algorithm to input the sequence and process and then develop the structure, and you have to submit the structure. And they assess us this competition will be held once in 2 years, and then it will be run for 4 months. And for each per week, they will give you a 10 or 5 to 10 targets. It depends, it may vary. Okay, so they will compare with the actual solution, and then they will declare who is the best predictor in categories, several categories is there. So protein structure prediction, model quality evaluation, etc. Okay, so these are the worldwide competitions. Next slide. So actually, in 2016, I have developed a model quality assessment method, that's SVMQA, and then participated in this competition, along with me, 47 different methods also participated in this competition. And among these 47 methods, SVMQA is the top. You can see here on the right hand side, figure A. You can see my method was at the top of the top among the method participated in CASP 12 experiments. And yeah, so I was invited to give a talk in CASP 12 competition in Italy. And in this the assessor they have to evaluate, they have to describe about the each method performance. In the conclusion, Andre, he mentioned that single model methods excelled in picking the best models from decoys, with particularly impressive results demonstrated by the newly developed SVMQA and ProQ3 methods. So this is performance of SVMQA. They highly prized in this paper. And next slide. So unfortunately, there is no competition for sequence-based peptide therapeutic functions. So I got an opportunity to write a review article in medicinal research review last year, 2019. So this is the title of the talk, title of the paper, like a machine intelligence in peptide therapeutics, a next generation tool for rapid disease screening. So actually, I use this as a platform to evaluate the existing method, like a CASP-like evaluation. I wanted to do like a CASP-like evaluation of the existing method. So I did that. Next slide. Critical evaluation of sequence-based peptide therapeutic prediction methods. Next slide. So basically, I cannot consider all therapeutic peptides. So for this evaluation, I consider only like 6 different peptide therapeutics, like anti-cancer peptide, anti-hypertensive peptide, anti-tubercular peptides, anti-inflammatory peptides, quorum sensing peptide, and cell-penetrating peptide. And you can see here the positive sample is like very small, but the negative sample is huge. But no one has done this kind of evaluation. And then I want to make sure like each positive sequence should not be present in the existing method training data set. So I exclude all those peptide sequences, and then I applied a CD hit of like, if I have the large data set, I applied a CD hit of 0.8%. If it is a small data set, I applied a CD hit of 90%. Okay, and this is the frequency of the peptide of positive and negative samples. And here you can see the length distributions. You can see here the length distribution between a positive and negative sample for ACP and CPP is almost same. But whereas in case of other therapeutic peptides, the positive and length distribution is varied. Next. Here I have shown list of currently available ACP predictors. The first column is that predictors. Second column is classifiers and year, data set size, feature encoding, evaluation strategy, accuracy, and web server availability, etc. So to date, 16 methods for ACP prediction. Actually, this is based on 2019, like December 2019. So 16 methods of our ACP prediction have been developed. Like a 40% of the method use the same data set, and 67% use the same classifier but different input features. The remaining methods used a slightly larger data set and different classifiers. Actually, you can see here some methods. You can see here IACP method in this figure, like in this table, IACP method data set, you can see 138,206. The same data set utilized by Khan et al. And other method like IACP, GANC. So actually there are several methods. They developed their prediction model, but they did not evaluate their model whether it has a transferability or not using an independent data set. So basically they are taking someone else's data set and then they're over optimizing and then show the higher accuracy, and to show their higher accuracy, they just over optimize the prediction model and then they are reporting. So actually these kind of method have limited practical application. So in the future, like if someone is interested in developing a prediction model, please try to check your model transferability using an independent data set. So here I have selected all the web server that is publicly available, and then I assess their performance using my newly constructed validation data set. And this is the list of currently available CPP predictors. And these 3 are anti-hypertensive peptide predictors. And these are all the 4 different anti-tuberculosis peptide predictors. So 4 methods are available for anti-inflammatory peptide predictors, and then this is the QSP predictors. You can see here in the QSP predictors, this is the first method proposed by Professor Raghav group. You can see here this QSP pred FL and peptide suit. These are all published in briefings in bioinformatics and bioinformatics. Although they published recently, like 2018 and 2019, still they are using the same data set that proposed in 2015. Even they are not. They did not attempt to update the data set. Why I'm saying because data set construction is very difficult, as I mentioned earlier, and it needs a lot of human resources. Next slide. Next slide. Next. 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 Okay. Here we have shown the performance of ACP prediction method using an unbiased data set. So we have used 246 ACPs and 1,733 non-ACPs. The first column represents the method employed in this study, and second column is that Matthew correlation coefficient, and the third column is that balanced accuracy. So you can see here that we have used like a imbalanced data set, 8 times higher than the positive data set. The negative data set, the non-ACPs is like approximately 8 times higher than the positive data set. So here ACC is not a straightforward metrics to compute the performance. Therefore, we used balanced accuracy, which is nothing but half of sensitivity and half of specificity, and area under curve, and then p-value. So here the methods are ranked according to the balanced accuracy. So among the existing method, MACP pred remains at the top with an accuracy of 87.6%, followed by the Professor Raghava model, that's anti-CP model 2. And you can see here this ACP pred FL ranked at the last. Although the authors have used a rigorous method to develop the prediction model, they over-optimize the prediction model. As a result, it is showing like closer to, it's not like it's worse than the random prediction. Even like if you predict all as a positive or all as a negative, then you will get a 50% accuracy. And see, you can see the anti-ACP pred FL performance is like less than the random prediction. And the p-value is like a comparison of this top method with the other method. You can see except anti-CP model 2, MACP pred significantly outperform the existing methods. Next slide. So here I have shown the all performance of all methods on different peptide therapeutics. ACP methods I have explained. Next is that antihypertensive peptide. So the antihypertensive peptides, like we are doing like much better. This our method MHT pred is significantly better than the rest of the methods. Whereas in the ATP, anti-tuberculosis peptide prediction method, this anti-TB pred M4 performance is much better than the other methods. And AAP pred method, peptide suit is doing better than other method. In quorum sensing peptide, the first method, QSP pred SPM is doing better than the other methods. In the case of cell penetrating peptide, MLCPP is performing better than the other methods. Next slide. So actually these kind of evaluation is helpful to the experimentalists to pick the best model, and then they can do the calculation. And also like computational biologists, they can do like they can address the shortcoming of their own method. So among the ACP, like we participate, our method participated in 5 different groups: ACPs, AHTP, this anti-tuberculosis, and AAP and CPPs. So we did an excellent job in anti-cancer peptide prediction. That's the MACP pred and MAHT pred and cell penetrating peptide. But our method for anti-tuberculosis peptide and anti-inflammatory peptide is is worse than the best method. So actually we have several shortcomings of the existing method. So we will address in our future studies. So actually in future research, focusing although models have been developed by several groups, at least like some groups should focus on critical evaluation of the existing method of different peptide therapeutics using a standard data set is highly recommended. Basically, this kind of evaluation will be beneficial to experimentalists and also computational biologists. Next. Next. Yeah. So in addition to the peptide evaluation, like we have also like introduced several feature capturing significant information between positive and negative sample by different approaches like feature representation learning. Like feature representation learning, from the peptide sequences, we will extract several feature information, and then using those features, and we input it to the several different classifiers, and then we will take that predicted probability and also like a class label. All those information will be concatenated, and then it can be used as an input to the new input to the machine, and then to develop a more reliable model. Next. This is iterative feature representation learning. So you can see here, this is we introduced for DNA, DNA-based like epigenetic prediction, and also DNA replication origin prediction. So for given DNA sequence, like we extracted like 9 different feature encodings. And then for each feature, we optimized using F-score plus sequential forward search. And then finally, we'll get 8-dimensional predicted probability value. This 8-dimensional predicted probability value inputted to the support vector machine, and again, you will get one more predicted probability value. This predicted probability value incorporated to the 8-dimension, and we'll get the 9-dimension. And then again, this procedure will be repeated, keep repeating, and then you will generate like 67 feature dimension, and then we will see which feature dimension is giving the best accuracy. So that will be selected as the best model. So actually this is like similar to the deep learning approach. So this we have done using support vector machine. So in MACP PRED, like how to handle the imbalance. Next slide. How to handle the imbalanced data set. In MACP PRED, I have shown I did not address the imbalanced data set. I just make my data set as a balanced. Okay, so that I develop the based on the balanced data set, I develop the prediction model. So in this study, like in pro-inflammatory peptide prediction, we have shown like how to handle the imbalanced data set. You can see here the positive data set has like 666 pro-inflammatory inducing peptides. And negative data set have 1,916 non-pro-inflammatory inducing peptides. Basically, we generated 10 balanced data set. So each balanced data set has 666 same positive peptides. And then negative data set, it will be randomly selected the same number of like let's say like balanced data set have 500 pro-inflammatory inducing peptides. And then we will randomly select 500 non-PAPs from the negative data set, so that we constructed 10 balanced data set. And then for each balanced data set, we input it to the random forest and develop their prediction model. So basically, we used 5 different composition, and then we got around 50 prediction models. So all these will be fused, and then the predicted probability score is again optimized, and then to classify whether it given sequence is PAPs or non-PAPs. So this is the one way to handle the imbalanced data set. There are several other ways out there to handle how to handle the imbalanced data set. Next slide. So this is my summary. That's a key to successful machine learning based model development. Accurate data set construction is the first and most important steps in machine learning based prediction model development. There is no general approach for development of machine learning based model. Let's say like if someone is developing like some approach, that approach may not be fit to your research. Like it is always recommended to apply at least 3 to 4 approaches, that what I have shown, like iterative representation learning, or feature representation learning, or adaptive feature representation learning, or meta predictors, or stacking framework. So you can do like a 3 or 4 different approaches on the same data set and select the select the best one. Generally, it's hard to pick the best classifier for the specific problem. For smaller problem, if it is less than 10,000 samples, so support vector machine, random forest, these are all the traditional classifiers have an upper hand over deep learning. In the case of a larger training data set, deep learning algorithm is always recommended. So in addition to the classifiers and approaches, exploring feature encoding algorithms also plays one of the key role for the performance. Therefore, we recommend to explore as many as feature encoding during prediction model development. So in addition to the exploring feature encoding algorithms, so feature optimization is also recommended. The above steps can be very helpful for the development of robust model. Next. So I would like to take this opportunity to thank my collaborators, like Professor Guang Li, Professor Jiong Li from Korea, and Professor Heo Lin, Wei Chen, Li Wei, Konzo from China, Professor Kurata from Japan, Professor Kunihiro Kojima and Mahati from Japan, and Vachara from Thailand, and Zhang Zhou from Texas University. And this my I would like to thank my funding, National Research Foundation of Korea, and thank you for your attention.
Thank you, Dr. Bala, for a very informative and detailed talk. Now session is open for the questions. Please turn on your audio and everything, then you ask question.
Good morning, sir.
Good morning.
Good morning, sir. This is Anvesh Pandey from BBAU. Sir, I have a very basic question. Like I am a beginner in machine learning and deep learning and things like that, Python scripting in biological sciences.
Okay.
So what if I, sir, ask you that if I want to start doing things like peptide and all that you are doing, so from where should I begin? Like I have to begin from scratch. So what that scratch should be for me?
Okay. I suggest you take some paper.
Yes, sir.
Okay, and try to reproduce. Like for example, like people always like whenever they do the modeling, like advanced modeling,
Yes,
they start with some basic features and then like a basic classifier. Okay, so you select the same data set.
Yes, sir.
Since you are a beginner, it's very easy to compute some composition-based features. Or like there are several programs are available, like from Jiang Ying Sang group. Even Professor Raghu also have like a feature computing some script like a he's having some program like to compute the feature that's publicly available. You put the data set, even if you don't have the like a coding experience, you take that and then put it you give you a sequence and then compute the feature using those program. And then using scikit like if you spend like a week or a month, you can like a very base of this scikit, and then you input those features and then develop the prediction model. You can develop the prediction model if your data set is like less than 200 or 300. It's very easy to develop a prediction model using your laptop.
Yes, sir.
Okay, and then get a taste of like machine learning, how it's doing. Yeah.
Yes, sir. Thank you, sir. Thank you so much, sir.
Yeah.
Anvesh, I suggest to take one of paper of Manavaran Balachandran and try to reproduce. So wherever you find difficulty, you can always send him an email and get some help, and then you can run in this way.
Yeah, you're always welcome. Yeah.
Thank you so much, sir. Definitely, sir. I'll try that way.
Yeah, sure.
Take any of H paper and try to reproduce in the same way, using the same methodology. You try to repeat all the work, and wherever you find difficulty, you please contact him.
Yes,
And I
sir,
request Dr. Bala, you please help him, because this is a workshop where we are going to teach students how to do certain things. Okay. Anyway, any other question anyone have? Okay, if not, then let's thank Dr. Manavaran Balachandran for his talk, and thank you, Bala. Thank you.
Thank you so much, man. Thank you.
Now it is time for the break. We will be having 15 minutes break, and then the talk will be given by Professor Kunal Roy. Now we are going for tea. Thank you.
Hello, Dr. Srivastava. So this is Kunal Roy from Jadavpur University. So I'm fine. So the time of my presentation is 11:45. Yeah, I'm ready. So I cannot hear anything. That's why. Yeah.
Welcome everyone. Welcome to the post-break session. In this session, we have Professor Kunal Rai, who is a professor and head of the Department of Pharmaceutical Technology in Jadavpur University, Kolkata. He has several awards and fellowship, including Commonwealth Academic Staff Fellowship, Marie Curie International Fellowship. His Scopus H index is 43 and total citation is around 10,000. So you can understand that he is highly cited and he is very successful. He has developed several methods and models for quantitative structure activity relationship. He is editor in chief in molecular diversity, which is an international journal. And he got several funding and several awards. Let us welcome Professor Kunal Roy. Now it is your turn. Thanks for coming.
Thank you, Dr. Srivastav. Can you hear me?
Yes.
And can you see the screen? Can you see the screen, full screen?
Yes, it is also.
Okay, so I can start then. Okay, good morning to everybody, all participants. This lecture is on some software tools developed at the Drug Theoretics and Chem Informatics or DTC laboratory. So first I will give some introduction to QSAR or quantitative structure activity relationship. QSAR is basically a statistical approach of ligand-based chemical design. I will use the term chemical design because QSAR is not only used for drug design purpose, it is also used for design of chemicals for modifying their properties or activities or toxicity. So we can use QSAR for diverse purposes. QSAR relates to quantitative structure activity relationship, but we can model property and toxicity also. And in those cases, the corresponding terms will be QSPR and QSTR, quantitative structure property or toxicity relationship. Anyway, the basic principle remains same for any kind of QSAR or QSPR or QSTR analysis, that the response property of chemicals will be modeled using chemical information. Now, chemical information is derived from chemical structures, and sometimes physiochemical properties or some molecular features also can be used as information about molecular structure. And this information is actually converted to some numerical form, because as I've already mentioned that QSAR is basically a statistical approach. So we need some numerical data for model development. So the structural information and information about molecular structure are converted to numerical form, which are called descriptors. And descriptors can be computed using different algorithms at different levels. So at the simplest level, it may be like you say molecular weight or log P, or it may be some 3D information also. So there are different levels of calculation of descriptors. You can use physiochemical derived or experimentally derived properties also as descriptors. So we can model biological activity, which can be therapeutic or toxic, of diverse kind of chemicals, including drugs, toxicants, or environmental pollutants, using descriptors with the help of some chemometric tools, which are basically statistical tools. So the basic notion of any QSAR model is that biological activity is a function of chemical structure and or property. Or we can say that biological activity is function of descriptors. Now, if we change the structural feature of a series of molecules, we can make some changes in their side chain or some substituent pattern. We'll see their activity differs, and actually the pharmacological class also may change. For example, say these are the 3 examples of compounds of phenothiazine categories. The first example is chlorpromazine, which is an antipsychotic compound. Second compound is promethazine, which is antihistaminic compound, and third compound is ethopropazine, which is anti-Parkinsonian compound. So all of them are phenothiazine derivatives with some changes in the side chain and the substituents in the ring. And we see with these changes, the pharmacological category changes. So this kind of changes, the impact of changes with changes in the biological activity, in their duration of action, and the agonism, antagonism, etc. Changes of biological activity with stereochemical features, including geometrical isomerism, optical isomerism, conformational isomerism, etc. So this kind of study, if it is done in a quantitative fashion, that means the changes of activity with changes in structural features can be studied in a quantitative fashion, because these changes do not occur in an abrupt fashion. They occur in a systematic fashion. So the relationship between molecular structure and changes in biological activity in quantitative fashion is the center of focus of any QSA study. And as I've already mentioned, that it is not only activity that we can model, but also we can model any kind of property or toxicity values. So any kind of response, which is dependent on chemical structure and or property. For example, so when we talk about biological activity is not only the interaction of a particular drug compound with the receptor or the target site, but also its behavior in the biological system through different processes like absorption, distribution, metabolism, and elimination. Now, all these steps have their own kinetics, which are actually dependent on the molecular structure information and their different physiochemical properties. So all of them can be actually modeled. So those also come under the purview of QSA. Then different physiochemical properties, say the pK value, and then the log P value. These are measured property, but they are also dependent on chemical structure information. So we can also model them. The pK is a very important field of research in QSPR. For compounds, if we do not have the experimentally determined pK value, using the appropriate model, we can predict the pK value of the drug candidates or any kind of chemicals. Similarly, QSR can be used in different other fields like, say, food sciences. It can be used in agricultural sciences. It can be used in nanosciences. In case of nanosciences, it is very important to determine toxicity of nanoparticles. So QSR is an important tool for prediction of toxicity of nanoparticles and nanoparticulate compounds. And there are various emerging topics also, like, say, determination of mixture toxicity, like the compounds which are present in the environment, like different environmental pollutants. They do not appear in their isolated form.
Professor, I'm sorry to interrupt. Your slides are not moving actually.
Okay, so you cannot see. You are seeing only the first slide then. But
only we are seeing the first slide. So are you talking only on first
slide, or
you have moved? So it's not changing for some reason.
Okay, so I can again unshare and share freshly in that case.
Maybe that will be better.
Can you see the slide now? The new slide.
Yes, sir. The slides are visible now. We can see.
So you can see a new slide now, no?
Yes,
so,
sir. QSAR.
The previous one, I'll show that.
No, we are seeing only the first slide, not the. Now we can see the second slide. Yes. Now we can see the second slide.
Okay, this is actually the second slide, which I have already discussed. The QSR as a function of chemical structure and property, which is presented in the form of descriptors, the numerical depiction of chemical structure information, and the examples I gave here, like 3 phenothiazine compounds. You can see the phenothiazine compound structures here. Are you able to see the phenothiazine compound structures?
Yes, I see now.
Okay, now it's okay. Okay, so these are the 3 phenothiazine compounds: chlorpromazine, promethazine, and ethopropazine, belonging to the classes of antipsychotic, antihistaminic, and antiparkinsonian that I have discussed. And this was the slide that I was discussing. That is the QSR is actually the broad term. We can include both the structure activity and structure property relationship in broad sense, and it is an intellectual exercise of assembling, manipulating, and examining data obtained from physical, chemical, and biological, and also computational experiments, and correlating the response or property using chemometric tool with the chemical structure information. This I've already mentioned about QSPR and QSTR, where the response is property or toxicity, then we can use the term QSPR or QSTR. This is basically ligand-based approach, so we need to start from molecular structure representation. We have to represent a molecular structure in an appropriate way, and there are different ways of representation. It may be represented in 2D form or 3D form. And correspondingly, we can calculate molecular descriptors which will have information about the molecular structure. And then we can use some statistical tools for feature selection. Followed by feature selection, we can apply some fitting techniques for development of models which may be regression type or classification type. Now, regression type models are used when you have a continuous biological activity value or continuous response values. But in cases you do not have the continuous values, you have only graded data like say active or inactive, toxic, non-toxic, or highly toxic, less toxic. In those cases, we can go for classification-based strategies. But the descriptor values will have their quantitative value. So classification models are also QSAR. Now, as I mentioned that from chemical structure, we can derive different descriptors, different classes of descriptors, and we can calculate descriptors at different levels. So we can actually derive from molecular structure thousands of descriptors. So we may have a very big table. Now, table looks like this. So different columns will correspond to different descriptors, and each row will correspond to a particular compound or observation. So we may have a big table which is called QSAR table, and one of the columns will be biological activity. One or there may be more number of columns also for biological activity. So from this table, we have to apply some data reduction tool. That means all the columns may not be important, and because some of the columns may have some constant values of descriptors, and some of the columns may also be intercorrelated, so they may carry the similar type of information. So to avoid redundancy, we have to omit some of the columns by removing constant value descriptors, intercorrelated descriptors, and so on. And descriptors which might be less important for a particular response, those also can be removed. And after data reduction, you can use the trimmed version, the smaller version of the descriptor matrix for feature selection. Feature selection is a method by which we are able to know that which features are actually important for the particular kind of response that we are modeling. Now, if we change response from particular activity to other activity, or from particular property to other property, or from one toxicity to other toxicity, the required kind of features will always change. Although the descriptors have information about chemical structure, but each of the descriptors have some physiochemical meaning, or they actually correspond to particular type of feature. So we can view descriptor in terms of feature also. So it is not only the particular type of group that we are discussing or that may be important, but it is the feature that is important. So based on that required feature, we can also extract the required combination of features for a particular type of response through the application of the feature selection tool, and then we can apply appropriate regression or classification strategy to develop the model, where we can have an equation like this, and this equation that what we call as model is then used for checking its quality in terms of its robustness and performance for the training set compounds, and also for new set of compounds for validation purpose, because the model that we develop will ultimately be used for new set of chemicals. So it is important to for us to know how the model will perform for the new set of data, and that is obtained through a process called validation, and we usually use a separate set of chemicals which have not been used for model development. So they are new to the model, but we know their biological activity or response values. So this knowledge is important because we'll apply our model for prediction purpose. So we'll have some prediction, but we need to know whether these predicted values are close to the observed values or not. So if we have the experimental values for those compounds, then we can compare the predicted value to the experimental ones. And for a good model, the predicted value will be close to the observed ones. That means the difference or residuals will be less. So that is a diagnostic test to know whether a particular model is predictive or not on a new set of data. And once we are satisfied about the performance of the model on new set of data, we can apply it for new set of data for prediction and for mechanistic interpretation. So the model contains some combination of descriptors, and as I've mentioned, that descriptors carry chemical information, and these chemical information are actually indicative of some kind of features, physiochemical features, which are important for biological activity. So these features can explain the type of biological activity or type of toxicity or property that we are modeling, and they can help us to explain the mode of particular kind of action or toxicity. And this model can also help us to design new set of chemicals by selecting appropriate features which are important for the particular kind of response, or if the particular kind of feature is important for toxicity, then we can remove those kind of features to reduce toxicity. In that way, we can fine-tune the property or toxicity of a chemical so that we can have a new chemical which may have the predicted desired response. So this we can do in silico so that we can avoid synthesis of large number of chemicals, and finally we can synthesize and test only selected number of chemicals which have higher potential to be more active or less toxic. And in that way we can avoid synthesis of large number of chemicals, we can avoid time, manpower and money. So basically QSR is an in silico screening tool. As I already mentioned, the QSR may be classified in general to two types, regression and classification. Regression is used for continuous quantitative response data, which may be linear or a non-linear type. And in case of classification, we use the categorical data for the response. And we can use different tools like linear discriminant analysis or logistic regression and so on. For regression, the simplest method is the multiple linear regression, and the more robust one is partial squares, which is actually a generalization of multiple linear regression. QSR has several steps, like data preparation, processing, prediction and validation, and interpretation. So let us see in some detail about these stages. So we should have a set of chemicals with a defined endpoints. So QSR cannot be started with very low, means limited number of sets. So if you have say 4 or 5 number of compounds and if you want to do a QSR, that is not valid, because QSR is a statistical approach, and for statistical reliability you should have a large number of chemicals. The more you have, more reliable your model will be. So you cannot do your QSR with a limited number of chemicals. If you have only 4 or 5 number of chemicals with biological activity value, and if you want to do QSR, you cannot do so. So you should have a sizable number of chemicals. So you should have at least say 50 number of chemicals, or at the risk case say maybe 30. Now for biological QSR, usually the response values are converted to the log scale. This is because if you see the dose response curve, the dose response will show parabolic nature, but if you take log dose response, that is sigmoidal nature, and the middle portion is linear, which is easier to model. So that is one reason. And second reason is that if you have your biological activity value in terms of concentration. Why concentration? Because for QSR we need dose fixed response type of data. Dose fixed response type of data means the dose required to elicit a particular response. This is because these are multi-point assay, so which is more reliable instead of fixed response fixed dose type of data. So if you have data at a particular dose or concentration, that is a fixed point, which is less reliable. So we need multiple point assay. using that is dose fixed response type, like say EC50 or IC50 or LD50 and so on. So actually, in biological activity, we use concentration, the concentration required to elicit a particular response. So here, what is varying is the dose or the concentration. And this concentration should be converted to log scale, because otherwise there will be the wide range of values, say 10,000 or so. So if you have such a wide range, wide span, then the that is a matrix that you calculate, say the simplest matrix is say R square. So I'll explain it later with the expression. So if you use R square, there will be the range is important actually. So for biological activity scale of a wide range, if you use the, let's say, 10,000 range, they will always be predisposed to a higher value of R square or other metrics, so which do not actually reflect the actual quality of the model. So you need to scale it, and you have to convert to the log data. So you have to use log response data or log concentration, but we need here log 1/c, because as the response is fixed and concentration is varying, so for a particular response, a compound requiring lower concentration will be more active actually. So a compound that has the IC50 value lower is actually more active. So that's why we need 1/c, that is 1 by reverse of concentration. So we'll use log 1/c as our response in traditional biological QSL. So this is about the response part, and for the chemical structure or chemical structure information, we have to present the structure in an appropriate form, and we can compute descriptors using our appropriate algorithm. Now, if it is 3D type of descriptor, then we have several other steps before that. We have to do the conformation analysis, and we have to do energy minimization and so on. But if it is 2D descriptors, there is no need. We can directly calculate descriptors from 2D representation of molecular structure, or it may be simple 1D or 0D also. So then we develop our QSL table, and the first job will be data pretreatment, that is removal of intercorrelated variables, etc. And as I told, that it is important to know how a model perform on new set of data, and in many cases, the new set of data may not be available. So in those cases, what we do, we divide our available data set into two parts. One is called training set, and other is called test set. The training set is used for development of the model. The information that is present in terms of both biological activity and chemical structure information are used to for the training purpose to develop the model. And the test set compound is kept as unseen. This is never used for model development. The model has not seen the test set information, so it is new to the model. So we can use this model as a check of the predictive power of the developed model. So we develop the model from the training set data only from feature selection, followed by appropriate regression and classification tool. And when we apply the model on the new set, that means test set, then the factor that is important is applicability domain, whether the model developed is applicable for the new set of data or not. Because QSL is based on similarity principle. So it learns based on chemical features. So if some chemical feature is not present in the training set, the model developed cannot predict for that kind of feature. So it is of no use to comment that if a particular test set have some particular feature that is not present in the training set, so the prediction will always be bad because it is outside the applicability domain. There is no question of prediction of that particular test compound using a model that has not seen that particular kind of feature. And the developed model is validated using the variety of metrics for checking its predictive power. Not only predictive power, basic statistical quality of the model and robustness, those are also checked using different metrics. And finally, We interpret the results. We can use the model for designing purpose and prediction for new set of chemicals. As I was mentioning initially, that QSR is not only applied for drug design, for modeling activity or ADME properties or toxicity, for design and optimization of leads and refinement of existing drugs, but it is also used in material science for fine-tuning property to have some material with optimized property, for enhanced operation selectivity and optimized process efficiency. And it is also used in predictive toxicology, especially with respect to ecological context to model ecotoxicity. This is done for risk assessment, keeping in view of hazard identification, hazard categorization, and prioritization of chemicals. So what QSR is, basically, QSR is the application of biological activity or response knowledge of limited number of chemicals to develop a statistical model or tool which can then be used on a large set of data without experimental value for prediction purpose. So this large set of data may be hypothetical also, maybe virtual, may not have been synthesized yet. So we can have their predicted values even before synthesis, and based on the pattern of the values that we are getting, we can decide whether we'll go for synthesis and testing of those compounds or not. We'll only choose those compounds which have higher potential to become active or less toxic and so on. So QSR can be used for identification, optimization of leads, prioritization of chemicals, reduction of animal experimentation is important, economical and time-saving, exploring the mechanism of action, risk assessment of chemicals, and for sustainable chemical operation. Now, in the field of drug discovery, for drug discovery, it is a very lengthy process and time-taking. It can take 10, 12 years for a compound to see the market. So it starts from a very initial stage, that is discovery and development through target selection, lead finding, lead optimization, then identifying the routes of administration, pharmacological profiling, then preclinical research in animals, toxicity study, in vitro, in vivo, both short-term and long-term. And then based on that, there may be the investigation of new drug application based on data, animal data and manufacturing information. Then we can go for clinical trial in different phases. Phase 1 in healthy volunteers, phase 2 volunteers with disease condition, and phase 3 for large number of patients. And then comes the new drug application, review of new drug application, and then in some cases approval. But there is a large number of rejection cases in different stages, in different stages of clinical trial. So later it fails, so more is the cost involved. So if a drug has to fail, it should fail at very initial stage. And that's why the in silico technique is important, because if after phase 3 a drug fails, then you have invested a lot of money, manpower, and time. So it is of no use then. So if a drug has potential to have failed, it should be demonstrated at very initial stage. There are several successful applications of QSR along with other modeling techniques for drug discovery, like say zanamivir in influenza, tirofiban in thrombosis, imatinib in cancer, raltegravir in AIDS, donepezil in Alzheimer's disease, and so on. As I was mentioning, that QSR is not only applied for drug design, but it is also used for chemical hazard estimation. So as you know, that large number of chemicals are in the market, large number of industrial chemicals are there, and new chemicals are coming to the market at a very high rate. But we do not have sufficient data about their toxicity potential or hazard potential or their risk assessment data on different environmental species. So actually, for less than 1% compounds, we have adequate data. For the rest of the compounds, we don't have data or sufficient data. So in the absence of hazard data of these chemicals, we may use the predictive tools to have computed data about their toxicity potential. And in absence of experimental data, this QSR or QSPR derived data is useful to gap the information. With the ever increasing production of new chemicals and rising cost of experimental testing, and considering the requirements to optimize resources for risk assessment of existing chemicals, different regulatory authorities like US EPA and European Commission's Scientific Committee on Toxicity, Ecotoxicity and Environment recommend quantitative structure activity relationship as a reliable tool for ecotoxicological hazard estimation. So we can actually predict physical and chemical properties and environmental fate, ecological and health effect of organic chemicals in absence of experimental data. And as we have very limited amount of experimental data available, especially with respect to several environmental species, we have to use some kind of model or mathematical technique to have an idea about their persistent, accumulative and toxic or PBT behavior. And not only prediction, QSR can also be used to design safer chemicals, safer and greener chemicals for development of sustainable society. So due to increasing requirements for alternative methods to in vivo toxicity testing, a variety of in vitro and computational methods are being proposed, as I've already mentioned, by US EPA and the European Commission's Society for Ecotoxicity measurement. So under the new REACH regulation, it is more important to have this computed or predicted data to bridge data gaps. In QSR, in regulatory use, the QSR is very much effective in terms of time and cost effectiveness for animal experimentation, to avoid animal experimentation, to support 3R principle, that is refinement, reduction and replacement of animal experimentation. They can also be used for virtual compounds, and QSR modeling is supported by various organizations like European Center for Validation of Alternative Methods, International Organization of Medical Sciences, USEP, and so on. Now, in the field of predictive toxicology, we can use different endpoints for modeling, like say inhalational toxicity, genotoxicity, nephrotoxicity, development toxicity, dermal toxicity, and so on. In material science, we can use different type of materials like fullerenes, catalysts, carbon nanotubes, ionic liquids, and so on. And let us see for some more details about QSR model development. Like say, we start with chemical structure and calculate descriptors. And then, as I mentioned, that we have to derive the QSR table, and this is followed by the appropriate feature selection tool and application of appropriate algorithm for regression or classification, followed by model building, and then we interpret, validate, and predict the model. Now, in case of regression model, if it is a very good model, the predicted compounds will be very close to the observed ones, and in that case, the residual will be less. And we'll see this the points are very close to the 45-degree angle line. The points are very close to the 45-degree angle line, so degree of scatter will be less. But in case of a bad model, these points will be scattered in a scatter plot. Now, if you see the historical development, actually the concept started long back. As we can consider in 1868, the Crum, Brown, and Fraser used this concept to mathematically correlate chemical constituent with physiological action in the context of stricting derivatives. In 1899, Mayer and Watson used that is partition coefficient to correlate the narcotic potency. In 1935, Hammett used benzoic acid derivatives to develop LFER model, linear free energy related model, to correlate the reaction rate with the substituent constant. Then in 1962, Harnsch applied QSR for plant growth regulator, and he used the same concept of Hammett. He modified the Hammett substituent constant in the context of lipophilicity to derive the hydrophobic substituent constant, or pi. In 1962, Free and Wilson used mathematical models. This is based on using some indicator variables, presence or absence of some descriptors. To develop a model, but that has limited application because it cannot be generalized. In 1975, Brandick and then in 1977, Keir and Hall, they explored the topological concept. They developed different type of topological descriptors derived from 2D representation of molecular structure. And then in late 1970s and still continuing the advancement of topological QSR. In 1988, Kramer introduced comparative molecular field analysis in the context of 3D QSR, and this was followed by that is Gomshia's comparative molecular similarity analysis. And then 1990 onwards, virtual screening studies various ligand-based and structure-based approaches, and 1997 onwards various multi-dimensional QSR formalism like 4D, 5D, 6D, and 7D. So if we see the dimensional aspect of QSR, we can see that 0D is chemical formula derived, 1D substructural fragment derived, 2D graph theory derived, 3D spatial geometry derived, 4D conformation, orientation, and coordination state representation derived. 5D induced fit parameters are included, the ligand-based virtual or pseudo receptor model. And in case of 6D, we also add the solvation condition. And in case of 7D, we use receptor structure information. So nowadays, QSR is used along with other related fields like pharmacophore, docking and structure-based design, team informatics approaches, and database mining for modeling and virtual screening purposes. As I've already mentioned, that the chemometric tools which are used for QSR model development are in general of two types: regression-based and classification-based. In regression-based, we have the method of least squares and partial least squares, and in case of classification-based, we have discriminative analysis and cluster analysis. And also the machine learning method, which can again be regression type or classification type. Like to say, in case of artificial neural network and support vector machine, these are becoming popular in the field of QSR and machine learning. Just 1 minute. Okay, so as I was discussing about QSR model development, there are some requirements of the response data. The response should be the dose fixed response type. The concentration should be measured in a molar unit that I've already mentioned. The molar concentration should be converted to a log basis, log 1 by CLPC. There should be a span of at least 4 to 5 log units in the response data, and there should be sufficient data points are available. So actually for regression-based, classical regression-based problem, there should be at least, for each descriptor, at least there should be 5 data points. So this is a minimum requirement, but this may not be relevant in case of the more robust techniques. So if we see the simplest case, there's a multiple linear regression, which in which case the equation looks like this. You can see the left-hand side of the equation you have the response, and in the right-hand side of the equation you have the descriptor values. So x1, x2, x3, these are the descriptor values, and a1, a2, a3, these are the corresponding regression coefficients. And after development of the model, we can plot actually the observed versus predicted. And as I've already mentioned, for a good model, the degree of scatter will be less. And there are different metrics to judge the quality of a QSR model, like determination coefficient, which is actually R square. So here if you see the expression in the numerator, that is sum of square of the residuals, the observed minus calculated whole square. And in the denominator, you see the observed minus y bar square. Here the reference is the mean of the training set. So obviously this denominator will be higher value than the numerator. And as I was mentioning, why we require to take log value, if you use the original raw data, this denominator value will be high, very high actually. So the R square will have propensity to become higher value. So we need to use the log converted value for QSR. But this expression doesn't say us anything about the number of descriptors used in our model. So if we use more number of descriptors, actually the R square value tends to increase. And if you use same number of descriptors as the number of data points, you will have the value of R square as 1. But that doesn't mean that you have a good model. So you have to penalize the model with the number of descriptors used. So if P is the number of descriptors, you can penalize it, and the R square will be a lower value than R square because you are penalizing for the number of descriptors. The number of descriptors should be lower, number of data points should be higher. The variance ratio or F value, that is actually indicative of the significance of your beta coefficients or regression coefficients and overall significance of the R value. Standard error of estimate, that value should be low for a good model. For a classification-based model, you have the metrics like sensitivity, specificity, accuracy, and precision calculated from true positive, true negative, false positive, and false negative values. Now, OECD has provided a set of guidelines for QSR model development. These guidelines were developed in 2004, actually, originally in 2002, then modified in 2004. There are 5 points. These are defined endpoint, an unambiguous algorithm, a defined domain of applicability, an appropriate measure of goodness of fit, robustness and predictability, and mechanistic interpretation if possible. Now, if we see the QSR steps in the light of OECD guidelines, we'll see that we start with OECD principle 1, that is the endpoint data. Actually, the endpoint should be such, as I've said, defined endpoint. So we can not use data from 2 sources with 2 different experimental. In that case, for the same compound, you may have 2 different values using 2 different experimental protocols. So we cannot use 2 data sets obtained from 2 laboratories who have used 2 different protocols to derive the experimental values. So we can merge this kind of data. Then OECD principle 2, that is the unambiguous algorithm. So whatever you're doing, that is molecular structure, representation, descriptor generation, then training test, division, then application appropriate tool for regression and classification, and prediction for test data, all comes under OECD principle 2. According to OECD principle 4, there should be appropriate measures of statistical quality and robustness and predictivity of the model. So you should use a variety of metrics for checking quality of the metrics model. According to OECD principle 3, you should check the applicability domain of the model, because the model that you have developed cannot be applied for the whole universe of chemicals. There should be defined applicability. For example, say you have developed a model for phenol toxicity, you cannot use it for aniline toxicity because you have only used information for phenols or different phenol derivatives. You cannot use it for amines. It's like that. So there is some chemical domain. And OECD Principle 5 says there should be mechanistic interpretation if possible. So that's why you have to use always interpretable descriptors. Now we should consider some points to consider QSR model development. The data should be homogeneous. There should be inappropriate endpoint data should be avoided. Use of collinear descriptors should be avoided, especially in case of classical type of QSR. Incompressible descriptors. Some descriptors may have been derived from some matrices or some mathematical operation, but may not have any physicochemical meaning. Those should be avoided. Error in descriptor values. So we use different software for descriptor calculation. So it may so happen that the software cannot handle some particular feature or some particular atom, especially say for organic metallic compounds, it cannot handle particular atom type. Or you are using the compound structure which are say enolizable or some resonance type. So you are using particular tautometric form for a particular compound, but for other compound you are using some other tautometric form. So the descriptor value that you will calculate will depend on the type of particular chemical feature that you are feeding. So it should be kept in mind. So it may lead to an error in descriptor values with different structures or different structure representation. For example, say nitro group can be represented with different forms. So accordingly, the descriptor values will also different. Then transferability of QSR. The QSR you develop should be transferable, and I should be able to reproduce the same one, which may not be possible always in case of 3D QSR. Applicability domain should be defined. Unacknowledged omission of data points should be avoided. In some cases, say some data points actually lead to poor models, so we omit those without reason, assigning any reason. That should be avoided. Use of inadequate data. I told QSR is statistical approach, so more data we use, better it is. So we should always use adequate and more number of data for QSR. Replication of compounds in the data set. If you are using some say some source, some data source, some database, the same compound may be used in two different names. So replication of compound in the data set should be avoided. Narrow range of endpoint values. If you have very narrow range, say 1 or 2 log unit, the difference between the higher and lowest active compound in your set, so that set is not suitable for your QSR model because there is no variation in the activity in the log scale. Overfitting should be avoided. Use of excessive number of descriptors in the final model should be avoided. Lack of inadequate statistics. Statistics actually help us to understand how good or bad the model is. So we have to use adequate statistics. Incorrect calculation should be avoided. A lack of descriptor autoscaling. Autoscaling helps us to understand the relative importance of different descriptors. Misuse and misinterpretation of statistics. Usually QSR models are developed by non-mathematicians. So the misinterpretation of statistical metrics or tools should be avoided. Consideration of distribution of residuals is very much important to understand the type of error. Inadequate training test selection, failure to validate a QSR model correctly, and the mechanistic interpretation is important finally. As I mentioned, that validation is a very important step for QSR model development to know whether the model will be applicable for new set of data, and there are different ways or methods of validation like internal and external. In case of internal validation, we use the same set of data used for model development for prediction purpose. Using leave one out and leave many out. In case of leave one out, you omit one compound in one iteration and we predict that compound using a model developed from rest of the data. And in that way, we iterate. In case of leave many out, we can take out some percentage of compounds in each iteration, and we can finally calculate the metric. The metric is Q square. You can see this formula looks like R square, the similar. Only the thing is that Y predicted is the leave one out predicted or leave many out predicted value. But the formula is actually similar. Bootstrapping is another internal validation tool, which is a sampling technique actually where the same compound can be repeated. And Y randomization is a method where we can actually randomize the Y column, keeping the X matrix intact. We can redevelop the model, keeping the same descriptor matrix, same descriptor combination as in the final model. But it is also possible to go for process randomization by selecting new descriptor combination at each iteration. But the most important is external validation, where you use a new set of data. We apply our model to predict the new set of data, and we can use this metric predictive R square or Q square external. Here also the formula is similar, only difference is that we are using here the predicted value of the test set compound. And in the denominator, we are using the Y bar of the training set response as the reference. So these are the things that are explained here. One more important thing is the training set selection, because the training set should be representative of the whole set. As it is a learning process based on similarity principle, all kind of features should be present in the training set. If some kind of feature is present in the test set but not in the training set, then the model cannot perform well on the training data, because there is no training for those features. So that's why if a compound with unknown activity is represented by a point in the descriptor space which is close to at least one point representing the compound with known activity, the prediction of activity of that compound is possible based on similarity principle, but otherwise it is not reasonable. There are different methods of division of the data into training and test, like statistical molecular design, Cohen's self-organizing map, K-means clustering, Kennard-Stone, Spears exclusion, all are developed based on similarity principle, chemical similarity principle, but one can also use the random method of division if one is using multiple division. And finally, applicability domain. For prediction purpose, one should check for applicability domain, because without applicability domain, a new compound cannot be tested by a particular model. And there are different methods of checking applicability domain. One important method is using the leverage approach. Leverage values are actually calculated from head matrix, from head diagonal. You have the leverage approach, leverage values, and the critical value can be calculated by 3p by n, where p is the number of variables plus 1 and n is the number of objects. So if the leverage value is higher than this critical value, then the particular compound is outside limit. But there are different other methods also for checking applicability domain. Now, if you compare the internal versus external validation, then it's a great debate actually. Some group of authors feel that internal validation is better because we are not omitting some compounds as a set out that is to keep set aside. So we are using data for all the whole set of compounds, and we are not ignoring any information in the set present. So if properly done, that can give a well-developed and validated model. But according to other group, the external validation is the only way for checking the quality of the model. And if you do not have any new set available with you, then the original dataset is divided into two parts. One is training, other is test. Test set compounds are not used, never used for model development. The model has not seen the test set information. So it is better to use both internal and external validation. And external validation is the gold standard for checking the reliability of a developed model. Now we come to the tools that we have developed at Drug Theoretics and Chem Informatics Laboratory. These tools are all QSAR-related tools for doing different operations for development of QSAR models. There are some smaller tools also for different specific operation, like say division of the data set into training and test, like feature selection from genetic algorithm. Then if you have developed model and you have the predicted data, then you can use a particular model for calculating different validation metrics, etc. Then some say stepwise model development and say modelability of a data set, etc. These are some of the smaller tools, and then some of the advanced tools are also there. Now these all tools are available from these two sites, and that these tools can be downloaded from this site free of charge. So some of the smaller tools like say MLR plus validation, dataset division, external validation plus. External validation plus can be used if you have the predicted data from any model, maybe MLR, PLS, or any kind of other, maybe some machine learning derived model also. If you have some predicted value, then this tool can be used for checking the quality and reliability of the model. Then base subset selection, genetic algorithm, bias variance estimator, etc. So here I will discuss about some tools like AD using standardization approach. So as you know, the applicability domain is very important, and it's important to know whether a particular model is applicable for a new set of data. As I told, there are several tools for applicability domain. It may be say based on leverage approach, or it may be based on range-based approach, maybe based on PCA approach, etc. So here we check the applicability domain using a standardization approach. Now a particular model is developed based on a combination of descriptors, and the training set compounds will have the corresponding descriptor values for all these compounds, for all the training set compounds. And we can consider that these descriptor values are normally distributed. And as you know that the normal distribution pattern, if you take the mean plus minus 3 times standard deviation, that covers the 99.9% of the population. So most of the thing. So anything that is beyond that 99.9% may be considered that outside the domain. Based on that concept, what we do is standardize all the descriptor values for all training and tested compounds. So these are method of standardization, the value of descriptor minus mean by standard deviation of the corresponding descriptor. And you get the standardized value. So if the standardized value is less than 3 for all the descriptors, that here we are actually calculating the maximum value, maximum standardized value. If it is less than 3, in that case, all the descriptors will have standardized values less than 3, and we can consider that the particular compound K, that is a query compound, that is actually within the domain, if it is yes. And if it is no, then we have to see whether the minimum value, minimum standardized value, minimum, there will be different descriptors, so we have to calculate the minimum standardized value. If it is more than 3, then all the descriptors are actually out of range. Then we can consider that particular compound is outside domain. But the problem arises when some of the descriptors are having less than 3 and some others having values more than 3, the standardized values. Then we have to apply the Z distribution, and we calculate this the mean plus 1.28 times the standard deviation. Now if this new value it is the more than 3, then we will consider that particular compound is outside domain, and it is less than 3, then inside domain. So this has been applied using different data set, and we have seen, so this method works well, similar to the leverage approach. So this particular one is actually the published in Geometrics and Intelligent Laboratory Systems with different data sets. This has been explained. Next one is double cross validation. This was also published in Geometrics and Intelligent Laboratory System in 2016. Now, as you know, when we develop a model from a training set of data, the training set is fixed. So as that is a fixed composition set, so it has some bias in descriptor selection. So to avoid that, we can apply the method of double cross validation. Now, what we do in the usual process, we divide the data set into 2 parts. One is training and the other is test. Now, in the conventional approach, the training set is used for model development, and the developed model is used for prediction of the test set compound. But in double cross validation, the training set is further stratified, k-fold, k times. So we have k calibration sets and k validation sets. So from K calibration sets, we develop K number of models, and these K models are assessed for their predictive performance on the validation sets that we have developed internally. Now we can select the optimal model. We can use different criteria, either based on the mean absolute error of the validation set or from consensus predictions. Consensus means more than one model. We can use, say, simple average of those predictions from the top model, or finding the best descriptor combination from the top model. So these are the three ways, and then we can use the optimal model, which this final model or consensus model can be used for prediction of the tail set. So this is the total double cross validation cycle. So it has an internal loop and also an external loop. So it actually avoids the bias in descriptor selection and validates the model in a better way. Next is intelligent consensus predictor. This was published in Journal of Chemometrics in 2018. Now, if we develop a particular model, we have some prediction, but a particular model may have some shortcomings. So with respect to some descriptors, with respect to applicability domain, with respect to mechanistic approach, mechanistic explanation, and it has a fixed combination of descriptors. So we are losing some information because you have not used some other descriptors which are not selected in that particular model, but those are also important. So it is always wise to use consensus prediction, that is more than one model. Consensus predictions are always more reliable than single model. Now, consensus means usually in QSL literature, the average of model predictions are used. But in this particular case, we are using intelligent consensus prediction. Now, what is intelligent consensus prediction? Now, here we are not using all the models for making average. We are using only qualified models. Now, what are qualified models? Now, qualified models are defined based on query compound specific way. Now, for each query compound or test compound, we have to see whether a particular model is qualified or not. So how we do that? For each query compound, we check 10 close compounds in the training set, and we check how a particular model can predict those 10 most close compounds for a particular test set in a leave one out prediction. So we take a cross-validated prediction error for those close compounds. So in that way, we can actually compare all the models, and in some cases, it may so happen that when we are choosing 10 most close compounds, we can see that some compounds are not that much close to a particular compound. In that way, we can have different criteria for choosing how much close a particular compound should be to take into consideration of this particular feature. And if you see that we cannot have at least 3 compounds as close enough to be considered, in that case, a particular model will be considered as disqualified. That means we will not particular model with the corresponding training compounds which are not that much close to a particular query compound, because the model is developed based on the compounds which are too distant for a particular query compound. So in that way, the model will not be suitable for prediction of that particular query compound, and the model is disqualified. And finally, we take an average of only those qualified models instead of all the models available. So that is called CM1, the average of prediction of all qualified independent models. Now, what is CM2? CM2 is based on weighted average prediction. Here we also use the cross-validated prediction error data. So some models will have lower cross-validated prediction error, and some models will have higher cross-validated prediction error. So we can actually that is, you use some weighing factor based on that cross-validated prediction error. A model having lower cross-validated prediction error will have higher weightage. And in that way, we can calculate the weightage average prediction in CM2. And CM3 is base selection of the model prediction based on query compound specific way. That means we select only one model per compound. Say, for example, for query compound 1, say model 3 is best. For query compound 2, model 2 is best. In that way, we do that in query compound specific way. And finally, we have the prediction for all the compound using specific model-derived predictions. And we can also use some additional criteria like Euclidean cutoff and applicability domain and Dixon Q-test. We cannot discuss it in that much details here because of paucity of time. And finally, this model-derived prediction from CM1, CM2, and CM3, and also CM0, that is the traditional average prediction. We can calculate the MA 95% value, mean absolute error 95%. Now, why 95%? In some cases, it may so happen that in a set of query compounds, most of the compounds are predicted very well. Only 1 or 2 compounds are not predicted well. They may have very high residual. So to obscure the capacity of a particular model to predict for most of the compound, we can avoid the high residual compounds provided that they are less than 5% of the total number of query compounds. So we try to identify those models which perform well for most of the compounds in the query compounds. So at least 95% of the compounds in the query set. So that's why we prefer MA 95% as a better metric than MAE itself. Okay, so we can then see the performance of different consensus prediction and individual model-derived predictions. And it has been seen that in most of the cases, the consensus predictions, intelligent consensus predictions, especially CM2 and CM3, perform better than the individual model-derived predictions. This is another tool, that is prediction reliability indicator. So when you have a model, then you have always some data, but it is important for us to know what is the reliability of prediction. So if you have already the experimental data available with you, then you can judge just from the residual. But if you do not have the experimental value known, in that case, you have to use some tool. So this prediction reliability indicator gives us that information. So what is the reliability of model-derived predictions for a compound for which you do not have the experimental value? So this was published in ACS Omega in 2018. So this is based on 3 criteria. One is scoring based on mean absolute error of closest 10 training compounds. So for each query compound, we choose 10 most close compound in the training set, and we try to see mean absolute error, leave one out for those 10 compound based on that model. Criterion 2 is scoring based on applicability domain of the model based on standardization approach. This is criterion 2 because applicability domain is important, and according to criteria 3, scoring based on proximity of the observed response mean to the training set, because actually the model that is derived from method of least squares, they perform well for compounds which are close to the training set mean. So this is another criteria we use, the proximity of the observed response mean, proximity of the predicted value to the observed response mean of the training set. So these are the 3 criteria, and then we use some weighing the combination W1, W2, W3, and get the composite score. And we can vary these scores W1, W2, W3, and we have seen that in many of the cases 0.5, 0.5. This combination works well actually, and although we get 0 for applicability domain, but as we are using these scoring based on mean absolute error of the closest 10 training compounds, so that information is inbuilt in here, and 0.5, 0.5, this weightage factor performs well. And based on that, we can give a composite score, CS3 for good prediction, CS2 for moderate prediction, moderate quality prediction, and CS1 for poor or unreliable prediction. So we can have an idea about the quality of prediction based on this tool. Now the final problem we have, that is the QSR model development from small data sets. In many cases, we have very small set, and it is difficult to develop a QSR model because we have to divide the data set into training and test sets. And as you know, the set is already small. Now, if you divide it into training and test, then what happens? The training set is further small. So we have very limited information for model development. So that is not desirable because we are losing some information by keeping aside a portion of the data set, test set. And secondly, if there are some outlier compounds, they can significantly influence the model quality in a small set. And also, as the composition of the training set is fixed and is very small, there's a bias in descriptor selection. So there are several factors of error in case of small data set. So how we can tackle those? The problem of model development from small data set can be overcome by double cross validation. So what we do, instead of dividing it into training and test, we do double cross validation. We divide into calibration and validation set. So we can redevelop model from different calibration set. So the fixed composition of training set, that factor is nullified, and we can have unbiased selection of descriptors. Finally, we can also use the consensus model prediction. So we can use actually more than one model. So we are using more number of descriptors actually, and applicability domain also is widened, and shortcoming of one model can be overcome by the other model used. So consensus predictions are always preferred for modeling small data set. And it is also important to do chemical curation, because if there is a problem, especially in case of small set, if there is a problem in the chemical structure supplied, so that will influence the quality of the model very heavily, because your set is already small and there is error, so that will significantly influence the quality. So there are several sources of error. The structure may be wrong, the normalization of bond is incomplete, some duplicates may be present. Other possibilities may be wrong charges, presence of explicit hydrogens in the hydrogen diffusion structure, incorrect bonds, and so on. So we have a 9 workflow which one can use for chemical curation purpose for removing these organometallic compounds, because in QSI we usually use the organic compounds and we do not use the organometallics. The mixture should be removed, and then the salt form is usually converted to the original acidic or basic form, and there is the optimized geometry, optimal normalizer, etc. And finally you get molecules in the SDF form. So there is a 9 workflow available in our site. Now for small dataset modeling and small dataset curation, we have two different tools integrated into one, that is small dataset curator and small dataset modeler. In case of small dataset curator, after chemical curation, what we do duplicate analysis, structural outlier removal, response outlier removal, and activity cliff determination. Now duplicate, the two inputs having the same chemical structure and same values of descriptor are actually duplicates. But if you have the biological values different for those two compounds, if the values are very close, then we can take average. But if they are significantly different, then we have to omit both of them, unless we do not know which value is correct. Then one can do the outlier analysis in terms of the response range. It may so happen some of the compounds are having value which are too much different from the rest of the compounds. So if this is an outlier, that should be removed actually, because that will otherwise significantly influence the model quality. So this we do based on this. If the value of the response is outside the range of mean plus minus k times standard deviation, the k is usually 3, then that particular compound can be removed. Then the outlier analysis based on the structural pattern. So we calculate the descriptor values, and we calculate the Euclidean distance scores, and that is standardized. That is within the value of 0 to 1. And then we calculate the mean distance scores. If this score is outside the mean plus minus k times standard deviation, then we can omit that particular compound, because in that case, that particular compound will structurally dissimilar from rest of the compounds. And then finally, activity cliff analysis. Two compounds having similar structure or close structure, but they are having quite different values of the biological activity, then there is possibility of presence of activity cliff, which may not be handled by the QSR model developed. So to identify activity cliff, how we do that, that is based on these three criteria. So it depends on the number of similar compounds. If the number of similar compounds is 3 or more than 3, then we have to apply criterion 1. If it is 2, then criterion 2, and if it is 1, then criterion 3. If it is 3 or more than 3, then what we do, we take mean value of the similar compounds and we compare it the value of the query compound. So mean value of the similar compounds minus response. These should, if it is more than threshold, the threshold is usually 1 log unit, and if the corresponding t value is significant at p that is 5% level, in that case there is possibility of activity cliff. If the number of compounds is 2, then what we do, we compute mean value of the similar compounds and we calculate the difference from the response of the query compound. If it is more than threshold, and also if it is more than the difference between two similar compounds, in that case there is possibility of activity cliff. And finally, if the number of similar compounds is only 1, we will just see the response of the similar compound, the difference between response of similar compound with the response of the query compound. If it is more than threshold, that is 1 log unit, then there is possibility of presence of activity cliff. So these are the different stages are applied as a part of a small dataset curator, and then comes small dataset modeler. Now, in case of small dataset modeler, let's say we have n number of curated compounds, and we can use R out of n compounds in each validation set. Let's say R is the number of compounds in each validation set. It can be user defined. And we can actually prepare calibration sets and validation sets. So there will be several combinations. Actually, R can be taken in different ways from N. So let us say as a K number of calibration set and K number of validation set. So from K number of calibration set, we can develop K number of MLR or PLS models. And then we have to select the top MLR and PLS model based on different criteria. We have these 5 criteria in our tool. That is model with least MAE 95% of the validation set, a model with least MAE 95% of the modeling set, model with the highest Q-square leave 5 out modeling set. Then we can use also pooled descriptor from top 3 model based on the least MAE validation set. And also we can use the consensus model, simple average and weighted average of the top 3 model. So we can select one model, and then finally we can choose the final model based on this, which can be the single model or some consensus model also. So here we do not recommend for going for training test division. Instead of training test division, we can do in this way by double cross validation. So this is the thing is written here. That is, we are calculating these different metrics for different iteration, both for internal and external validation in the framework of double cross validation. But finally, we choose a model based on the MAE 95% value. We prefer the mean absolute error as a measure of choosing the particular model instead of the metrics like R square and Q square, etc. Because R square and Q square metrics are biased, they may not reflect sometimes the quality of a model because they are based on some ranges and comparison with mean. So we will always prefer the error-based data. So these are the GUI for the small dataset curator and small dataset modeler. This was published in Journal of Chemical Information and Modeling in 2019. And so far we have discussed about different tools, but those are actually tools for different stages of QSR model development and validation. But recently we have introduced one more tool, that is DTC QSR. That is a complete QSR modeling tool which incorporates different smaller tools into a single tool. Of course, we have not incorporated tools like intelligent consensus prediction or prediction reliability indicator in these tools yet. So we can do it afterwards, but this is a complete tool for development of regression and classification based model. So it develops both the regression and classification based tools. All steps of visual model development and validation are performed using the same software. Variable selection is done by genetic algorithm and base subset selection. Modern modeling techniques are MLR, LDA, and random forest. Applicability domain is done using standardization approach. Performs wide randomization test, and there some plots are also generated. And there is a page for frequently asked questions to handle those software. And if still there are some additional query, there is a form given here. One can fill this form with the snapshot of the problem one is facing while using this software. So all these software are downloadable from the sites I've already shown. And to run the software, Java should have been installed in the system. So these are the two web address from where the software tools can be downloaded free of charge. So to know more about QSR, those who are new to the field should have the preliminary training in the field of QSR. So this is a book from where one can understand the basics and principles and practices of QSR. It is published by academic press. And this is a more that is a shorter version. Is a quick for quick learning about QSR and QSPR as Springer brief. And this is a more reference, the reference book on QSR for application in drug design, predictive toxicology, and risk assessment. And this is another reference book, advances with application in pharmaceutical, chemical, food, agriculture, and environmental sciences. And for more specialized application, like say in Alzheimer's disease, multi-target drug design, then drug repurposing, and ecotoxicological QSR. Some more references are there. Two more recent additions, but these two are not yet published. The in silico modeling of drugs against coronaviruses and chemometrics and cheminformatics in aquatic toxicology. So those who are interested can follow these books. And thank you very much. If you have any question regarding QSR, I'll be happy to answer. Thank you.
Thank you, Professor Rai, for detailed talk. Also for So now the session is open. Yeah, if any of you have any question, you are welcome to ask. Okay.
Sir, you're on mute.
Sir, hello?
I cannot hear you.
So, yeah.
So anybody is asking any question? I am hearing some questions, but very low volume. Okay, if there are no questions, then let's thank Professor Roy for his very informative talk on especially on 2D QSAR. Now at 2:15, his group will be taking a demo session to explain all the tools and techniques and softwares developed in Professor Roy's laboratory. So whoever is interested to perform 2D QSAR and want to use these free tools, which are freely available from his website, they are welcome to attend this session. And all the hands-on participants, they should join this session at 2:15 after the lunch break. Lunch break now, and thank you, Professor Roy, for coming. Thanks a lot.
Thank you very much.
Am I audible, sir?
Yes, you are audible and you are visible. We have 2 more minutes.
How to share my slide?
After introduction, you can share the slide. If you want to test, you can share it now.
How to do that, sir? Can you help me, sir?
We'll call you. You mute
Yes.
your microphone. You mute here. Good afternoon, everyone. Welcome to the post-lunch session of 6th day of International Workshop MMTT 2021. In this session, a group of Professor Kunal Roy is going to explain about their tools and techniques, whatever they are using for QSAR analysis. So Kabruddin Khan is there to take the hands-on demo, and you are all welcome to interact with him and interrupt him whenever necessary. So Kabir, now it's your turn. You can start.
Thank you very much, sir. Just one basic question. All here are from medical chemistry background or anyone from informatics background who have worked in computational chemistry before?
So we have participants from several different backgrounds, and it is live on YouTube for future. So anyone can see this, any background.
Okay, to start with, you've already seen the presentation of my guide in the morning. So this I will explain to you all with the examples. Okay, so to start with, the title I have the same title what sir has used, software tools developed at the Theoretics and Informatics Laboratory. This QSR, as we all have seen, it deals with the development of the model, which correlates biological activity with chemical descriptors. Here, descriptors are the functions of the properties, some properties of the chemical. And there are several geometric methods available in QSAR, which are further classified into types, classification types, and machine learning. Among regression types, most familiar and common ones are MLR, multiple linear regression, and partial least squares. Our classification, we have discriminant analysis, clustering, cluster analysis, etc. And there are some machine learning methods, artificial neural networks, support vector machine, etc. So today, in today's explanation demonstration, I'll restrict myself to multiple linear regression, partial least square, discriminant analysis, which is here linear discriminant analysis, and cluster analysis. So the rest two we will not cover in this today's presentation. Okay, these are some examples I have shown, some of the examples of our tools. I have mentioned named a few of them. And to use these tools, the first condition is that you should have Java installed in your system. Most of these tools are Java-based applications, so just to make sure that your system has Java installed. Okay. Okay. Now, regarding the software, so these softwares, as you can see, they are available on this particular website, ttclabs.web.com. Okay. I'll show you that. Can you see this page, all of you? Can you see this?
Yes, sir. It is
Yes,
visible.
The website available. You just have to go to this. I'll just show you how to go there. Just simply type lab, and you will find this one link, and which will open up into one website wherein all the softwares are present. As you can see, this is the DTC QSAR classification. These are all tools which are made on DTC by DTC lab members. And there is another website wherein the similar sets of softwares are available. As you can see on the screen.
sir, we cannot see on the screen. There is this black screen which is seen after the presentation.
Is it visible now?
Not yet, sir.
Previously it was visible, right?
Sir, the PowerPoint is still there. The front screen, there is a screen in front of it which is black, totally black.
Yes, sir. It is visible now.
Okay. Can you see this?
Yeah. Before we start, please make sure you have it's visible. Yes.
Yes, sir. The presentation is possible.
So this is another website wherein all the softwares are present, as you can see. So from these two websites, you can download almost all our softwares free of cost. There is no need of any subscription or any money payment or something. Purely free of cost. Okay. So I'll get back to my slides now. So as you can see, some of the names I have mentioned here: data pre-treatment, data subdivision, modified KMDI, genetic algorithm, etc., etc. And so, as I have mentioned, classification, like I have mentioned, linear discriminant analysis. For that, a single tool is available to perform the entire process of linear discriminant analysis, which is named as classification-based QSAR. And as sir have mentioned in the morning, there is something called as intelligent consensus predictor. So for that, we use intelligent consensus predictor, this software, and there is something called prediction reliability indicator, which is used to reliability of the external sets which are not used during the model development. And there is also a presence of complete package wherein named DTC QSAR. Okay, so this gives you the entire this in this software you will find the entire linear regression, that is MLR, plus the regression which is classification type in one tool. Okay. Okay, so there are some additional software we need when we are dealing with QSAR model development, which are not by our lab actually, but normal QSAR model development. The first one is 9 which is a software available free of cost online, and the link is also provided on the PTC Lab website. You can see here data that it is named as data curation. You all can see this data curation.
No, sir.
Is it not visible? Data curation.
No,
Sir, only
sir.
a black screen is visible.
No, sir.
Yes, sir.
So again, that black screen has come in front of the slides.
Can you see the browser now of my browser? Is my browser visible to you all?
Not yet. Not yet.
Can somebody from administration can help me for this? Hello?
Sir, if you can try not sharing, and then again share it, maybe because of that.
It's gone now?
Yeah, it's gone. And reshare your entire screen. The screen one, hash screen one will be there. You share that thing.
Okay. Can you see now this page of mine? Is it visible
Yes.
in the browser?
Yes, it is visible now.
Yes.
There is the data curation symbol you can see here. This is, if you click on this will directly take you to the website wherein you can download the NIME platform. So I have clicked it now, so it should open in minutes. So this will take you to the NIME wherein you can download the NIME platform, and additionally our lab has developed some process of some protocols for some methods like chemical curation for chemical curation. Some nodes are developed in our lab, and for biological curation. This website should take you there. So we'll start by the time it will open, okay? So I wish we all interact with each other when we are proceeding with the model development. Okay. So can you see this page of mine? Hello, can you see this?
So folders are visible to us. Biological data.
Yes. Okay, now listen to me carefully. Okay, so to start with any QSAR model development, we need data. We need an experimental data. The data can be an activity for a compound, activity for any target. It can be an activity for COVID. It can be an activity for Alzheimer, malaria, etc., etc. Or it can be an activity or toxicity for a compound. For example, compound X has got so much toxicity. So for any QSAR model development, the first thing what we need is the data. The data are in two forms. First is the biological data, and second is chemical data or the chemical structures. So there are some strict guidelines we need to follow when we are collecting data or when we are generating data. So I'll show you some examples. Here I have collected some raw data from a, can you see this Excel sheet which is opening? Can you see that? Hello, can you see this Excel sheet which is open now? Hello?
No, it is showing a small square, black square. Let it open.
Okay, now can you see this Excel sheet? It's visible to you all?
Yes.
So sample data I have downloaded from a website called Ecotox, which is a database for toxicity data. And as I have mentioned, for QSAR we need biological data as well as chemical data. So for biological data we need compound, that is structures, and they are reported activities. As you can see, there are some activities reported in the website and which unit, in which unit they were reported. Here it is active ingredient in milligrams per liter. So, and one more thing, the OECD guidelines, as you have seen in the morning presentation, there are some rules and regulations laid down by the OECD, Organization of Economic Cooperation and Development, so which says that you have to follow certain rules while collecting data or while executing model or while developing model. So for collecting data, you have to be sure that these data which are collected has to be performed or obtained using a same bioassay protocol. What it means, you cannot club data for two different bioassay protocols in general. There are some exceptions, like if there is a scarcity of data, there is no data available at all, and you have only limited number of available data. In that case, you can it still perform the modeling, but that will be not considered so much reliable because you have mixed the data from 2 or 3 different bioassay sources. So these I have taken solely from a single source, having a uniform bioassay protocol, and these are cache numbers of the specific compounds against which these toxicities are reported on the website. Now, what are the problems when we collect the data? As most of you are from medicinal chemistry background, you all will be generating responses in your own lab. So these problems you might not face in future. But if you wish to perform QSTR analysis of the data which is already available in literature or on database, or some lab is providing you for the experimental data. In that case, you have to make sure first they are collected, they are generated using the same bioassay protocol. Secondly, there should not be any duplicate. In this case, as you can see, when I have downloaded the raw file, there are number of duplicates available with different reported toxicity values. As you can see, for the same compound, you may find different values. Yeah, these three are the same compound having 3 different values. So one thing is to make sure there should not be any duplicate in your data. When you are developing a model, you should not take a same element twice with the 2 different activities. That makes no sense. You're saying compound A is having this response. Again, you're saying compound A is having some other response. So you have to be more sure which one you are taking. It is possible if your single compound can give you multiple response at multiple. That depends on individual error also. Like different people have different. So that comes. So, but for modeling, you have to be sure that you're not taking anything twice. So as you can see, there are other sheets also available here. I have separated the data here now. I have removed all the duplicates, like no compound is represented twice, and I have collected here around 144 compounds with their reported toxicity data. Okay. Can you see this Excel sheet now? Is it visible, this Excel sheet?
Yes.
Okay. So here you can see these I have extracted from the previous Excel sheet now, which is a more concise form of it, and this is their reported concentration in activity with uniform scale units. Okay. In the morning, sir has mentioned usually for QSR model development, we convert these values, the reported response values, into some concentration. Why we do that? Anyone remember what sir said? Anyone? Why do we have to convert concentration into some these values into some concentration? Okay, when we plot these values again, so usually the dose response curve, when you plot this values and all, you will get a parabolic or some other form of curve which is difficult to interpret. But once you convert them into a log form, they tend to give a linear line, which is more easily interpretable and can be easily understand for a normal person, and for explanation also it becomes very easy. Okay, so here I have compiled and converted them into a uniform unit where it is molars per liter unit followed by logarithmic conversion. So So this was the original value which was reported. So to convert into a molar form, it's divided by the molecular weight of the structure. Okay, and then after made it into a molar form, so it's divided by 1000. It was initially in milligrams. So for molar, it was divided by 1000. And finally, the log conversion, and these are the final values which I will be using as my final response. Okay. So is it clear so far now regarding the data treatment or data pre-treatment? Is it clear so far or anything I have? Any answer? Is it clear so far?
Yes.
Slide. Visible?
Not yet, sir.
Okay, now it is visible.
Yes, it is visible.
Yes.
So as I have said, the data consists of two things: biological data and chemical data. Chemical data here means the structures. Always before when you perform QSAR studies, make sure that your structures are free of errors. And most of the cases, what we do, how do we generate our structures? Either we draw them, or we are provided by some distributor or from where we are collecting the data. Thirdly, or we download from a database, because there are n number of databases available which provides you structures like PubChem is there, Zinc is there, NCI. There are number of databases available right now. Okay, so to be sure that what structures you are claiming, that same structure should have been drawn and saved in your system. Then only you can say that your model is reliable and can be used for further predictions or whatever it is. So as you can see here, I have downloaded from a website named Chemical Book. I'll show you some of the structures. And these structures are same which were present in those Excel sheets. As you remember, the first column of that Excel sheet contains cache number. You remember? Okay, so this is one of the structures I have. Yeah, one of the structure I have opened here. This is formaldehyde, and you have to make sure that it is formaldehyde. It cannot be something else for that particular case. Okay, because when we are calculating descriptors, which is constant term, it should not, like for different molecules, descriptor values will change. So when you are claiming formaldehyde activity, so you have to calculate formaldehyde descriptors only. Okay, so you have to make sure that all structures are same, and you have to always compare the structure integrity with some standard website. Like for example, you are claiming acetaminophen is having so and so response, so you have to make sure that acetaminophen structure is drawn properly. If it is drawn properly or not, how to compare that? Go to the standard website. How, where you will find that? You can find it on drug bank. Drug bank stores all the structures of all the drug molecules. So once you have compared with that, now you're sure, yes, I have drawn it correctly. There's no error in the structure drawing. So as you can see, I have all the structures pre-drawn and kept. Okay. Okay. Now, second main problem here is when we draw structure now, there is one major drawback. Uh, Structures should be named as you want to be them to be named of. For example, outside it is, as you can see, it is named as cache number 50000. But when you open this file in some suppose notepad, here this is the main column. Can you see this small window?
No, sir. Black screen is there.
Okay, can you see this notepad text file?
Yes, sir.
Okay, so now as you can see, when you open any software file like MDM mole file or SDF file in a text format, it gives like something like this. The first thing is the name of the molecule. You have to make sure the name what you have given outside should remain inside. Because what happens is when you proceed to the next step, which is the descriptor calculation, this becomes your identifier. If this is not properly labeled, you will not be able to identify for which compound these descriptors function. So you have to make sure that these two are named identical. Is it clear so far? And this is very crucial actually. Is it clear?
Yes, sir.
So now we'll proceed. Okay. Okay, now we have a software named as assigned file name as molecule name. This software is made so with the help of this software, you can do this in one go. You may have thousands of structures or you may have 10 structures. It's very difficult and time consuming if you go naming each one by one. Like if you have 1,000 structures one by one, it will take at least 1 week. So it's a simple tool developed here to simply, is it visible to all, this software?
No, sir.
How to show the entire system of mine? Can anybody knows? Every time I have to show you.
You should try sharing the entire screen and not one window at a time.
Is it visible now? Is it visible everything now?
Not yet.
No, sir.
Anyone help me how to share the entire window so that I do not have to ask again and again? Organizer, anyone is there? Can you help
Yes,
me?
yes. So our system ninja will help you.
Just tell me how to share
Yes,
the
yes.
entire video desktop
Please.
forever.
Please click on the share. Click on that share.
Okay. Close share tray. Close share tray, right?
Close, then again open.
Okay. Again open. And then
Now, whether you are getting the desktop option will appear. Desktop.
yes, desktop option. Yes.
Yes. Click on that.
Okay. Now it's visible to all.
Yes.
Okay. So when you click on this website, this GUI, small GUI comes, and you do not have to name each structure one by one. Simply, you just have to direct the directory wherein all your molecules are present. In my case, I have placed it in. So I have placed my all structures here. So this has to select Control A and open and give a directory. So I've already done this, but still I'll show you. Here I have saved it and just have to click submit. This small two-step process will name all those molecules outside whatever it is named. That same name will transfer inside also. So it will be a your compound title. I'll show you. Okay. Same name. So if you open this compound with notepad, you can see the same name is available inside also. So when we are calculating descriptor, this will appear as first column as an identifier. So you can say, yes, this is my compound and following descriptors belong to this particular compound. And this is very important. Okay. Next, in some of the software, another is open label software we might need sometimes, because uh, uh, open label is a software which is helpful in converting one form of the molecule into another. like extension can be changed. So most of the software what we use like this is a commercial software and all software for descriptor calculation, which accepts SDA format. So for converting that into an SDA file, we use OpenBabel as one of the options for converting. Any one of you have used OpenBabel before? It's widely used software. Anyone?
No, sir.
Okay. So to convert that, initially we have downloaded mole file from the website. So I wish to convert that into a SDA file. So the input format was mine. You have to give MDL mole and choose a directory where your molecules are. So and choose the molecule which is already being named, like which is outside and inside having been named identically. So you just have to choose all the molecules and which format you want to convert, just change the extension. So here I wanted to change in STF format and give a location where you want to save the file. So I want to save the file in this. Okay, right now I will not do this. After assigning the directory, you just have to click convert and it will convert within seconds. Okay. So now my files are ready in a combined form. I'll show you. It is opening. Okay, can you see this file? Is it visible? All of you?
Yes, sir.
Okay. So these structures I have downloaded, name changed outside and inside, and it is now compressed into a single file. Okay. Now, usually in our lab, we do 2D QSAR modeling, and which I will be presenting today. So for 2D, don't need to convert structures into 3D. If it is already in 3D, no need to worry. It won't make any changes to the descriptor calculation because 2D descriptor don't need 3D features. And second important thing what we do is we add structure explicit hydrogen. Like if your compound contains hydrogen, it should be made explicit. It should be visible enough. And finally, you have to save the molecule into 2D or 3D. It's up to you. But for 2D models, you don't need to. If it's an optional option, either you can keep the molecule in 2D or 3D. Okay, now we come to the next part of it, which is chemical structure curation. We have already seen biological data curation, wherein what we do, we check for duplicates. No compound should be present in duplicate. Secondly, all the compounds what we are taking for modeling should be in the same unit. You cannot mix a milligram and gram compounds together. Like one compound activity is present in milligrams per liter, other one is present in grams per liter. That is not, should not be, and cannot be followed. Okay? Not should not be, cannot be followed. That makes no sense. It has to be an identical unit. Okay. So similarly, for chemical structures, there are certain steps for the final curation. Okay. Now where we can go wrong? Okay. The first thing is we can go wrong in a duplicate process. When we download, as I have shown you in Excel sheet, to simply take that cache number initially and download the structure, you may download same cache twice if you have not checked the initial step. Second thing is, not all databases contain and correct structures, because what is a database? Database is a source wherein people upload certain data, but whether they maintain it properly, regularly, we don't know that. So we have to be sure whether it is correct or not. So in some cases what happens, for some faulty structures might be present for the same cache, or in some cases you might find salts of a particular compound, like sodium salts of something, potassium salts of something, or in some cases, for example, tetracycline and all, these are drug molecules, they are available in the form of a mixture, like for example, calcium salt of tetracycline. So you will find two molecules of tetracycline joined together by calcium. So for a QSAR model development, you cannot take a mixture. In a single file, there should not be two or more structures combined. It has to be a single entity. If it is a salt, the salt part should be vomited. If it is a sodium salt, sodium should be replaced by the following subsequent acid. It should be replaced by H. If it is a salt of some base, it should be replaced by an acid. If it is salt of some acid, it should be replaced by some base. So if it is a sodium salt, that sodium part should be vomited and should be replaced by H. Okay, now the problem arises here is usually for QSAR model development, we use number of compounds. We never use 10, 20 or 30 compounds. Sometimes it goes in thousands also. And it is very difficult to check each and every molecule one by one to presence of salt or mixtures or any other errors. Okay, so for that what we do, we use NIME platform. As I was talking in the starting, so this has not opened still. Okay, this is a software. After once you go to that website and download, okay, it is open now. You can download the entire NIME soft platform from this link. You can see this link. Just one reply, please. Can you see this?
Yes.
This link will take you to a place wherein you can from where you can download the NIME. Okay, and how does it look? The GUI of the NIME. This is how it looks. This is available free of cost, free of charge. Okay, only thing is you will not get certain workflow which we are using. This has been developed in our lab. These nodes have been developed in our lab, which are used on NIME platform. So there are two process workflow we have developed. One is for chemical curation, and another one is for biological curation. So why we use chemical curation? As I was talking about, the chemical structures, usually what we are dealing with, they are in hundreds or thousands. So we cannot check one by one. So these NIME nodes can help you solve that entire problem of of weeks and days and months in just a matter of half an hour or 15 minutes as per your experience. And how does it work? It's simple. Just read this, what is written there. Read STF file. So this node will help you to take up the file from where you are giving the input. This node will help you to remove the mixture. Like if a molecule is containing a mixture, so it will strip the one part of it. And this will convert this into some suitable format for a software to process it further. And if the software your structure is having any problem now, this will not go further. This will be vomited at this stage only. And then after this node will help you to remove the salt part. Like if it is sodium salt of something, it will simply delete the sodium part. Or if it is a potassium salt of something, it will delete the potassium part. I'll show you how to do this. Just a few examples. Simply just go to configure and you have to locate where your file are present. If you remember, with the use of OpenBabel, we have converted this SDF file. You remember? So just give open, apply, okay, and okay. So now this software has taken that molecule to process. I just have to, and you can check this file whether it has been properly executed or not. Simply configure, and you can give the location where you want to see. I'll make temporary folder here right now just for the explanation. Okay. And execute. If this goes green, that means the process has been executed properly. If there is some symbol like exclamatory sign or some, if it is not turned green, that means those molecules were not taken up correctly and there was some problem either with the software or with the molecule. But as we can see here, it has turned green. So this shows that software has successfully taken the molecule. How to confirm that? We have to save those files into some directory. Okay. You just save the file here. This is not the same file what we were seeing before. This is another file which is being taken up by the 9 platform. Okay. So as we know now that it has been taken by the software. So what is the next step in data curation? The next step is removal of mixture. So once the software has taken the molecule, your input part is over. Now you do not have to input anything as a fresh. Everything has been taken into consideration by the software. Simply just have to go right click on this, configure. How see, there are 3 options here. If there is a presence of mixture in your compound, either you can discard that compound completely, or if there is a presence of 2 different entities and you want the biggest entity to remain, so then you can choose this option. Like for example, as I have given the example of tetracycline, it comes in a salt form of calcium. So calcium plus 2 molecules of tetracycline. Okay, so this option, if you are selecting, it will delete the one part of the tetracycline and it will remain only calcium and a tetracycline. And if you just say apply and press OK, so now this has now executed. So now it is taking all the molecules and removing mixtures from all the molecules. So now you can see this has turned green, and now if you want to check whether this has really worked or not, you can simply save that file in your suitable location where you want to save. Okay. So we have saved the file here. 9. So I'll name this as mixture remove. Write the extension also. That's here. And let's apply. Okay. And execute. Now if you go to that folder, so now it has created another file. See, if there were any removal out of 170 compounds, if any compound was identified as a mixture, it will automatically it will not appear. But since we have chosen the removal of the smaller part, it will still remain in the entire list. As you can see, none of the molecule have been removed. All are present here. Okay. So now the next step is you have to generate RD kit, like make a molecule suitable for the further processing. Simply just have to click, configure, apply, okay, and execute. This step is not successfully done, or it will show you some error symbol like exclamatory sign or something. Then you can understand that this process has not been taken place properly. Now you want to check whether all molecules have passed the criteria or not, or there is some problem in some molecules. This will give you the passed molecule, and this will give you the molecule which has been failed. We will not check this. We will simply check if any molecule of R has failed the RD kit criteria. Simply give the location where you want to see that. So we will name this as RD kit fail. If there is no failure, means it is understood that all the molecules have passed. See, as you can see, the size of this file is 0 kb. That means there were no failures. All the molecules have passed this criteria and are fit for the further processing. Okay. Similarly, if I'll not explain each and everything, the process stands same for all the things. In this process, you can remove the salt. You have to assign the directory. First, you have to configure it and then execute. This step will remove the salt part of it. Like for example, if your molecule is containing sodium salt or potassium salt or some salt of some basics or salt of some organic compounds, it will remove the salt part. And the next step is usually for QSR model development, most of the software which are designed to calculate descriptors, they take up only organic molecules. And what constitute an organic molecule? They constitute hydrogen, carbon, nitrogen, oxygen, fluorine, bromine, iodine, chlorine, phosphorus, and sulfur. And when you deal with inorganic part, that is completely a different science in QSR itself. That is nano QSR and all. But in general, the available descriptor calculating software, they take up only organic compounds. Okay. So how this node will also help you to check whether your compound contains any of the inorganic parts. If your compound contains a mercuric part, so that makes your compound either organometallic or a completely inorganic salt. So that should be removed. And again, the same question, you cannot check that individually, opening each and every compound one by one. So this can be dealt with using this node. Simply just configure it and execute it. And in the next step, what will happen? Whichever organic compound will reside in this, and whichever inorganic or failed molecule, they will reside in this. And in the next step, since you have removed the salt part, you need to optimize your structure again. So you have to optimize here, and then you have to normalize, like you have to balance. Like if you have omitted the sodium part, it has to be balanced. If you have omitted some other part, it has to be balanced. So that is done using normalizing the structure. And at the end, you add hydrogen and you save the final molecule. In these basic steps, you can do a lot in very quick time. Some people they deal with in lakhs of molecules, and that entire process of chemical curation can be finished within 15 minutes. So any doubt in understanding these notes, you can ask now. I can repeat those parts. Anyone? Any doubt, anybody, or nothing understood at all? Just say yes or no. Am I audible? Am I audible to all? Hello? Hello?
Yes, sir.
Anybody, any doubt? I cannot hear anyone. If you have not understood anything, I can repeat, no problem. So those who will be pursuing PhD in future, they might be in this, they might need QSR for the model development. So if you have no doubt, we will proceed. Just say yes or no.
Yes, sir, no doubts.
Now, how this chemical curation is done with the 9 flowchart. Okay. Similarly, biological curation. In biological curation, first said in the morning, we check for the activity cliff or similarity. Activity cliff, when we check, for example, we have two similar molecules and there is a slight change at one particular position, the placement of hydrogen and the placement with something, the activity changes drastically. For example, bioactivity value is in 0.1 nanomolar and suddenly it changes to 0.001 nanomolar. Simply changes. These are, we call it activity cliff phenomena, and that can be checked here. So in those cases, we have already what to do with this molecule. Most of the people, they discard either both of them, or we using some principles they retain both of them. So that can be performed here. And this is this node can also be checked the similarity, how similar the structures are. So this can also help you to identify the duplicate. I'll show you one slide here. As you all can see this is how we identify the duplicate in the molecule. For example, if you have two different compounds with same distance similarity, you may think you may you can like you can check them for the duplicates whether they are identical or not. If they are identical, no need of using both of them. You can omit one of them. If you skip at the first step, like while data collection, for some reason you have missed to identify the duplicate. At this stage, using 9 platforms, you can identify and visualize actually, because same molecules will give you same distance similarity. See this. These two are identical. Similarly, these two are. So these compounds were having some data ID also. So their IDs were also same, a database ID. So we can see here. So far it is clear. Any doubt?
Yes.
Okay. So now now these people came from different backgrounds. People come from medicine, chemistry, cheminformatics, or pharmacoinformatics. When we are developing QSR models, we need the next half of it, which is a descriptor, molecular descriptor. They are functions of such structures. What is molecular descriptor? They are function of some chemical structure, meaning they are a constant value. They should not change whether a person is calculating them in India or in US or in Europe. Those values should not change. If the structure is same, they should give a same descriptor whatever it is, wherever you calculate it. So we have number of softwares currently which calculate descriptors. Some names I'll mention here. Okay. Does anyone of you have heard of pedal descriptor? This is freely available right now. Okay, one more question. Hello, does anyone have performed QSR before? Am I audible? Hello, am I audible?
Yes, sir. Yes, you are audible.
Okay, does any one of you have performed QSR before? Or any here? Yes or no? Am I audible? Oh, just say yes or no. It can be yes or no.
Sir, I have not.
Okay. So there are multiple softwares available on the available right now which can calculate descriptors of a chemical, and these descriptors range from thousands and thousands. Description can be many. So one of the very famous and freely available software is the pedal descriptor software. This is available free of cost at this particular website. Can you see this website? yaptwsoft.com. Name is pedal descriptor. So from this website you can download this latest version of this software, which is used to calculate the descriptor, and this gives you around 4,000 descriptors in total. So this is the latest version available for public. And how does it look in the? This is the GUI of the pedal software, and how to calculate the descriptor for those compounds. Simply just give your molecular directory where you have created the molecule. It's available on desktop. And next is there are multiple types of descriptors available in this. As one thing was sir was focusing mainly was that you should not use all the descriptors available. Only those descriptors which give some meaning to your descriptors that it can be explained properly which correlates with your target. For example, if you are studying toxicity of some Alzheimer drug in the brain, so in that case, it will be useful to use some statistical descriptor having no relation with toxicity of a molecule. Usually for toxicity to occur in the brain, we need a lofty profile. So in that case, you can go for a lofty calculation, or there are some other lofty calculations, or some other features like eta descriptors and all, and triple lofty. These are selected descriptors which can be used in certain cases, and similarly for other cases. If you're not working in Alzheimer's, some other targets, you may choose some other descriptor which is suitable for your target. Okay, so you can select whichever you need, and there are 3D also. So usually you should not use 3D descriptors, because for 3D descriptors, your molecule should be optimized and minimized, which we have not performed. So for that is another separate tutorial for that. So right now I have chosen all the 2D descriptor right now I have chosen for the demonstration, okay, and simply click start. Okay, this will start calculating your descriptor, but before that you have to assign where your descriptor will be saved. This is the output directory, and you can simply assign the location where you want to save the descriptor, and if you click start, it will complete the process and it will give you the output. And how the output looks like, I'll show you. So this is the descriptor I have calculated for my chosen compound, which I have showed you in the next starting. So the first column comes with actually this was processed already. The raw file contains a cache number as first column, and then comes all your descriptors. These are all the calculated descriptors which I have calculated. In my case, I have calculated this descriptor from two different software. One is not really available, but the process remains same. Okay, and other examples of descriptor calculating software are OChem. This is a web-based server which contains nearly all the available descriptors in the literature or in any website or something. I'll show you how it looks, this platform. This is the web base for that. Here you can put under this section, you can calculate descriptors. But you need to log in here, then only you can calculate. This website is also free of cost. They will not charge you anything unless you have to create an ID. And you can calculate almost every descriptor from this website. You do not have any paid software or something. Either this website or there is another option of Cradle. So I have already shown you this example. Okay, so once you calculate the descriptors, you have now a file ready. The descriptors are ready for you. Okay, so once your file is ready now, usually the Cradle and all, they have some drawbacks. Since we are not paying, blank. That should not be the case. You have to replace them with 0 or with suitable terms. And in none of the content it should be either 0 or some numerical value of that particular descriptor. So federal descriptor comes with this drawback. Then you have to identify which are those columns devoid of some values, and then only you can use this descriptor for model development. Okay. So what I have done now, this is my complete file of compound number identification. Like I have named those compounds with numbers. One is the compound number, and this is the cache number, and these descriptors they are much difficult to explain right now, because for them how are based on some calculations, some formulas, and which is not required right now. At the end, I have placed my activity column. This is the activity column I have placed at the end. And this entire column, this entire descriptor belongs to one particular compound. And and this activity is for same compound. Similarly, other activities for other compounds. So this is my final file ready to be used for model development. So far any doubt can ask me. Still descriptor calculation or chemical curation or biological curation. Any doubt? Anyone? Am I audible? Hello?
Sir, no doubt.
So now we have calculated a number of descriptors. So next step is normalization. We have to this is an optional step we usually don't need for regression type models. This is used in classification type models. So this I will explain in the next part when we are dealing with the classification model. Okay, and the fourth part is modulability index. This usually when we do is like if you want to check beforehand whether your model can be made or not, whether it is good to invest some time in developing a model or not. But usually these two are optional steps. Need not cover right now, and I will not explain them. Okay, we will directly move on to data pre-treatment. So what is data pre-treatment? When we have calculated descriptors from a descriptor calculating software, it will simply calculate all the descriptor which you have chosen and it will give you the output. But there is a problem. What is the problem? For example, we have two descriptors having the same values identical in both the columns. So we have understood that these two descriptors are not giving different information. So we may discard one of them. Otherwise, there will be a problem of overfitting. So, and additionally, another criteria is that your descriptors should not be highly correlated among themselves. If they are highly correlated among themselves, they are giving you same meaning again and again. So to handle, to tackle with that problem, we have another tool. You can see here. Wait. Okay. So this is the another tool. What is this? I'll tell you. So simply keep the correlation 0.99 and choose the input descriptor file and choose the output directory. What it does is it will check all your descriptors and intercorrelation among themselves. And if there is an intercorrelation of 0.99 or above, it will simply delete one of them and retain the another one. So it will reduce your pool. You will not have a multiple descriptor with same meaning. So this software can remove. I'll do it for you. Okay, I'll just choose the file and I'll locate it. Okay, I'll just name it as pre-treated file. If there is an empty column anywhere, it will show some error. As I said, there should not be any empty column, and here it contains some empty column. Just to identify that. See this. This was empty. That's why the software did not take this file. So we have to just delete, save it, and again run it. Keep the input file and make it 0.99. So now the process has started and job is completed. Your descriptor pool can be which however big, doesn't matter. The software will finish this very quickly. And now only at 0.99 correlation. Now initially it was around 700 something. Now it has vomited around 140 descriptors. Only 650 descriptors we have now remaining in the pool. So this has removed the intercorrelated descriptor, highly intercorrelated descriptor. So now we can use this file for further. The next step is the data division. Okay, so data division. Okay. So in QSAR, the identical norm says you have to place every fourth molecule into a test set. So what it means, 75% compound on an ideal basis in a training set and 25% of the total compound in the test set for the validation of the model. And for data set division, we have number of methods and tools available. One of them I'll show you here. The name is Dataset Division version 1.2. So this software will help you divide your data based on 3 different methods. One is Kenneth Stone, another one is Euclidean distance based, and third one is sorted response or activity based. These about 2 methods, Kenneth Stone and Euclidean distance based, they divide your data based on some distance method. Like in Euclidean distance, what it does, it will calculate the Euclidean distance of all your molecules. It will arrange it in ascending order, and it will place every fourth or every third or every second or every first compound into test set. Similarly, for sorted response or activity based, what it does, it will not take into consideration your descriptor values. It will simply take into consideration your activity values, the last column. And what it does, it will arrange it in hierarchy, either increasing or decreasing value, and it will place every first or every second out of 4, okay, or every third or every fourth compound into a test set. So let's take one demo and see how it is dividing the data. So just take the file which you want to divide. So I'm choosing this file, okay, and you have to assign the directory where you want to save the file. So I want to save this here. So I'll name this as seed1 and activity based. So activity based we have selected, and we will simply press start. Now, as you can see, here it is showing enter the number of compounds in each cluster. So what it is doing is, it is dividing your molecules into some clusters. What is a cluster? A cluster is a method wherein we group chemicals based on certain similarities. Okay, so here the similarities will be based on their Euclidean distance. So if we say 5, so what it means, if there are 10 clusters it has generated, so every cluster will have at least 5 compounds or 5 compounds in each cluster. So we have around 140 or 150 molecules. So we will give, in each cluster at least 4 molecules we need, because out of these 4 we know 75% should go into the training set, 25% should go into the test set. So when we give 4, it will generate that many clusters. Okay, and simply click OK. And now here comes the another window which says enter seed number. So what does the seed number specify? So for example, your software has calculated the clusters and it has sorted in a ascending or descending order. So when we say we want seed 1 to be placed in training set, so what it will do, every first molecule, count them, 1, 2, 3, 4. First molecule is placed in test set, and 2, 3, 4 is placed in the training set. Similarly, next 4. First molecule is placed in test set, 2, 3, 4 is placed in training set. This will repeat until entire list is finished. If I place this with 2, so what it means? 1, 2, 3, 4. Second is placed in test set, and 1, 3, 4 are placed in training set. Okay, right now we'll with this 2, and now it has completed. Okay, so now we'll see the output file, how it looks. See, this is the file it has given. As you all can see, training set is placed separately on this sheet, and test set is separated with this sheet. So manually, if you go to do this, it's practically not feasible, and you need software for this. So these software can actually help you divide your data using 3 different techniques: for activity based, Euclidean distance based, and Kenneth Stone based. I will not go each and everything, but this is only to show you how it performs. Okay, so far it is clear. Data division. Any doubt for anyone? So we will use this file for the next step. Next is model development. Okay, so we have now for model development. I'll show you one slide here. Everybody can see this slideshow. So now, okay, see this the 7th point, QSAR model development. There are 3 options given here: stepwise MLR, genetic algorithm, MLR best subset selection. So what are they? So far we have drawn the structures, we have curated the biological data, we have divided the data, we have pre-treated the data. But what to do with this data? How to develop a QSAR model? We cannot use all those descriptors to develop a model and say this is our model. It's practically nonsense. There is no point in it. We need to develop a model using the best features, which is showing some good correlation and it is having some meaning. So for that, we have number of algorithms. The most some of them are named here. First is stepwise MLR. I'll tell you what is stepwise MLR. So stepwise MLR, what it does, it will take all your descriptors and there is a activity column there. It will place your activity column. It will take one of your descriptors and it will see the correlation with your activity, R square value or R value. Okay, and if it is showing some good activity or good correlation, it will retain it. And in the next step, it will check another descriptor combinedly, or and it will check the R square value. If there is an increase of correlation, it will retain the descriptor, and similarly, it will go for the another descriptor. And this process will be repeated till all the descriptors have been examined. All the descriptors have been checked for the particular activity. For example, if you are working on Alzheimer, you have a response column with you, and there are 640 descriptors with you. So what it does, you have a compound X, suppose, tryptophan, imagine, and this is having a response value of 1.5 moles per liter. So we will take one descriptor, for example, it is log P value, and it will check how much correlation it is showing for that compound. And this will be for all the molecule, of course. And suppose one descriptor is showing R square value of 0.23, so it will retain that. In the next step, the next descriptor will be chosen, and in the next step, combine those two descriptors, R square value will be checked. And if it is giving better value, it will be retained. If it is not giving better, it will be vomited. And this will be repeated till all the descriptors have been checked against that activity. And all the best performing descriptors have been highlighted and given to you as an output. So what is happening in the background? We are selecting some features. So this step is also called feature selection step. And this is a very crucial step, and this step varies to individual. Like no two persons can get same set of descriptors, because we need to run this process a number of times, at least 8 to 10 times. And descriptors which are generated, we have to assess based on our judgment. Okay, so this is how the stepwise MLR works. Similarly, we have genetic algorithm. This is another method of feature selection. Genetic algorithm is like how genetic codes are chosen. In genetic algorithm, they select one parent descriptor and they see their correlation. And in the next step, they remove one of the parents and choose another daughter descriptor, and they check the correlation. And this step is repeated till the best possible combination of descriptors have been achieved. Again, this is a multi-step process. You need to repeat this process number of times so that you can get the best possible features available in your data. Okay, and this again has a drawback, like it depends on how many descriptors you have chosen. If you have chosen very limited number of descriptors, you do not have much option. If you have chosen a number of descriptors, you might have a better correlation with more descriptors. Okay, I'll show you how it is done with our software. Okay, so we have both the software, genetic algorithm and SMLR. So I'll show you with genetic algorithm. This is the GUI for genetic algorithm. And again, one more important thing is you cannot use test set for this. For feature selection, you should always use training set. And now see, you all can see the genetic algorithm, this UI with you. So now select the file, choose output location, and just write here GA. See here, now you can see here something, variance cutoff. This is again, suppose in the previous step you have forgot to do data pre-treatment, and you can do with this also directly. You can place intercorrelation cutoff 0.99, so it will not take two descriptors having this much correlation. And this is 100. What 100 means in iteration. So how many times that process will be repeated? I have said already. One descriptor is taken, check for the R square value. Second descriptor is chosen and check for the R square. How many times you want this to be repeated? So in ideal case, we see for the 100 times, and the best one are reported. And this is the most crucial step is the equation length. Okay. Now what the guideline says about equation length. So for an ideal model, the OECD guidelines lays down, it says for every 5 molecules, you can have maximum of 1 descriptor. And for 10, it will be 2, 15, 3, 24. So if it is 1000 molecule, you may have 200 descriptors. But that is actually, again, it cannot be followed. You have to make your model with the minimum number of features possible. So ideally it is 5 to 10 maximum. Above that, if you want to go, then your model doesn't qualify for best for, like your model is not so good actually. So in our case, we will keep it 4 for the simple reason. Okay, and just simply submit. Now you can see this process is running. So in the background, actually it is checking the correlation of each descriptor with the response value. As this will complete 100%, it will give you the output file. Okay, so now this has completed. Okay. See. Now this is here you can see so this has generated one model of yours, one best model using 4 descriptor, which is giving 0.51 as R square and 0.48 as Q. These are some validation metrics. I'll come to that later. So basically this has given you one best model with these descriptors: number of conjugation in a molecule, this particular fragment, single bond OH, and exposed role, and A log P tree. These are 4 descriptors. In the initial round, it has found to be the most correlating descriptor. So as I have said, this process should be repeated number of times so that we may get some better features and we can identify which are those better features are. So this has to be repeated at least 8 to 10 times. Okay, so this gives the output file also ready-made, so you do not have to search for it. You can simply copy if these are the best features, you can simply copy this and place on some Excel sheet. Okay, now the second step also has run. Again, in the next step, see, it has taken the same descriptor or different descriptor. So now in the second step, it has generated some better model. Initially, it was 0.51 R square, 0.48. Now it has generated 0.55 and 0.51. So there is a slight increment in the model quality. So this has to be repeated several times so that all the best correlating features are identified. And in some cases, it comes in negative values. Those models should be discarded. It should not be used. Okay. Similarly, we have a stepwise also for the same process. Again, the protocol remains the same. You have to assign your file and output directory and simply submit, and this will again give you some models. Okay. So this feature selection should be repeated until and unless you get some valid model. So that I've already done here, and I have identified some best descriptors out of that. So in my case, I have identified I've isolated around only 21 descriptors out of 640 descriptors. I've already identified and kept. These were the best possible performing descriptors I have identified in the data set. This can vary from person to person. You may get some better models than me also. Okay. So next step is so once we have identified the best possible descriptor, best correlating descriptors, what we can do is there is another software with us which is called as the best subset selection. So what it does, suppose you have identified around 16 or 20 or 25 best descriptors. Now you want to check the best possible combination in which it gives the best model. So simply double click on this, assign your training set, 2 different files, test set. Okay, how many descriptors you want? How many descriptor models? Like what is your equation length you need? So right now we will place around 6. Okay, and what is the minimum R square you want? 6, and intercorrelation we can keep up to 99. So what this software is doing is it is checking for the permutation and combination. What is permutation? NCN. How many types of combinations are possible for the particular set of descriptors? I'll show you in the output file. Okay, so now my files are ready. I'll show you how it looks. So now what it has done is it has checked for all the possible combinations out of those 21 descriptors in the pair of 6, all the possible combinations, and it has given me the output. As you can see, you can sort this. Okay, now we should also be aware about some cutoff values here. R square, the cutoff value here is 0.6, Q square is 0.5, and Q square of test set should be 0.5 minimum. But these are only a cutoff for an ideal and good model. It should always go above 0.65 or 0.7. In some cases, it goes up to 0.9 also. So those are exceptionally good models. So as you can see, none of our models are really good. The best model which is we are getting on 6 descriptor is 0.68, 0.62 Q square and other parameters, and this is Q square for test set 0.53. So what it means is only 6 descriptors are not capable of giving us a good model. So what similarly I have repeated this process for 8 descriptor also. I'll show you that. Okay, and now you all can see here there are some good models. At 8 descriptor combination, we have some some moderate or good models. See, there is a R square of 0.763 and 0.63. These are acceptable models and can actually be used. See, these values depends on your data, how good your data is. More the value, better the model, and you can expect the better quality data you have used. So this data is not so good, so it has given some moderate values, but still it is acceptable because it is over above all the threshold provided. Okay, so what we will do, we will take this model as a for final processing. So how we do that? Okay. See, what I have done on a single Excel sheet, I have placed my training set above and test set below. This is the response column, and I have regenerated the model manually. This you should not. This is I'll show you. Okay. I'll come back to this in the next step. So now we have a model has been generated. The next step is to validate the model. Okay. So how to validate the model? Okay. So this is our final training set having possible this descriptor what we have got in the best subset selection, and this is the test set what we have got in the final selection. And when we are giving any file to that, you always remember that the header should be same. There should not be any mismatch in the header. If there is a mismatch, the software will show some error. So we have got the descriptors, and now we want to see their validation metrics. Okay, so how to do that? Okay. Simply assign your training set. Set. Okay, so it has completed now. Now see the output file. Okay, so this is your model actually. This is how a QSAR model looks like. You have your response on the left hand side, and all this is your constant value, which MLR has some plus or minus deviation, and this is your coefficient of this descriptor, coefficient of this descriptor, coefficient of this descriptor, similarly so on. So this is your model. And which are the descriptor it has chosen? It has chosen 8 descriptor what we have given, and what they signify is they have some this is valence connectivity index, this is average molecular weight, this is eta index which signifies size, and this is another descriptor A log C which signifies lipophilicity, and number of conjugation which gives double bond and all, and so, and these are the metrics which we have obtained for that final model. Are you all following with me, or I am too fast? Hello?
No, sir, it's fine.
Okay. So as you can see, we were getting 71 R square. We can see here also Q square is 0.63, and similarly for external validation for external set here Q to F1 is 0.64 we were getting, and this is quality of the model based on MAE criteria, which is giving it as a moderate model. Okay, and there are some other criteria whether it is passing or not. These criteria are internationally followed, so it's called as Colbreth and Tropska criteria. These are some validation metrics. Whenever you develop a model, you need to show that your models are robust and can be acceptable at the international stage. So how to do that? Just show them whether they are passing whether different validation thresholds or not. So there can be different methods proposed by different groups, like Tropska group proposed some criteria, R lab proposed MAE criteria, or R square criteria. If those criteria are passed, you can say yes, my model is passing that criteria, so you should accept it. It is selective. One needs to show only the traditional criteria like R square, Q square, or some latest one which are followed. It is individual. It varies individual to individual. Okay. Okay. Similarly, for test sets, you can calculate the validation matrix using external validation plus. Okay. So again, here you need only okay. So this software external validation plus, this will give you validation metrics of only of test set, not of training set. So what it does, you just have to give test set file here, output location, and you have to give response mean value, like what it means, the mean value here, mean of this column, the response column, explaining this mean, simply average. So this becomes your mean of training set that should be placed here, and similarly range. How to calculate range is maximum minus minimum. This is the formula for calculating range, and this will give you all the metrics of test set validation. Okay. Now this was the validation part of it, and last and the crucial step is the applicability domain. Once we have developed the model, we have validated the model, it is passing all the criteria whichever we wish to report. Then we need to show what is the domain of the model, how big the domain of the model is. So when we say big, big matlab, how many, it depends on, like, first of all, the data size. If you have developed a model using only 20 compounds, 30 compounds, the domain is very small. If you have developed the model using thousands of compounds, so it has very large domain. And additionally, there will be a cases wherein your own molecules, sometimes they tend to fall outside the domain of the model. So let's check how it is done. Simply, you have to provide your training and test set for the calculation of AD. Give your training set, the final training set, what you have got, and then test set, and choose one output location. I'll name them as AD. Let's complete it. So in the final model of mine, in the training set, you can see there is one outlier detected, and this AD is based on standardized approach. So as already discussed about standardized approach, I'll repeat once again. In the standardized approach, you must all have heard about the normal distribution curve, a bell-shaped curve. So standardized approach, what it does, it assumes that most of the compounds will fall within 3 standard deviation range. So whichever compound goes above that, see this is about 3.09 or something. So that it will place that molecule outside the model. And similarly in the test set, it will check. So here we have it has identified two compounds as outliers. So what it actually means when we say it is outside AD, it simply means whatever model you have developed, it is not able to predict that compound's activity properly, or it is out of the scope of that particular model. Why it is important? See, when we develop a model, we develop a model with some intention. So we have developed a model, we have some query chemicals which is provided by some other lab. In medicinal chemistry lab, usually they work on some disease target and they develop a model, and some compound is provided to them and they want to check whether that model is within the scope of that model or not. So simply what they can do is they can calculate the descriptors of the query molecule, and they can check in spite of test set, they can give their query molecule as a subject, and it will show them either it is outside the AD or not. If it is inside the AD, the predictions which are done by the model, they will be reliable, and if it is outside the AD, you cannot rely on that prediction. It simply means that. There are multiple other methods for AD calculation, like Euclidean distance based AD or D-Modex based or H matrix based. There are many methods. In our lab, you will find Euclidean distance method based software, AD using standardized approach, and there is AD MDI. You can explore that later. So we have seen now applicability domain now. So with this, we complete the regression model development. So any of you have got any question or any query at any step, you can ask. If you wish me to repeat any part of it.
Yeah, the session is open for the question.
One more thing I would like to share. One more thing. Hello. Hello.
Yeah, please.
Okay, so actually, sir, I have not finished yet, but still I will try to minimize now.
So you have around 12 to 13 more minutes.
Okay, so I'll finish this in the last. This is the last offer I want to show it.
Okay.
Okay. So now this is the best part of it. So far, what I have shown you is in the graded manner. Like every single step, I have broken into individual particle, like data pre-treatment, model development, model validation, then applicability, domain analysis, and this becomes very time consuming and all. So at the end, our lab has developed a single interface wherein all this process can be finished within 5 minutes. Okay, so how it looks, simply have to give your input file. Okay, I'll use our previous input file. I'll use this file as input. It needs CSV files, so we have to convert this into a CSV format. Now you can see I have given, see, usually you need not follow the entire hierarchy of, if you know the basics. That was the reason I have explained you everything step by step. But this software, this tool will combine all the things and it will execute that thing in the background. You are not dealing them with directly. You are simply getting the output. Just locate the file. In the next step, you can select whether you want a regression model or a classification model. For that data what we have prepared is for the regression model, so we will be choosing a regression. Please listen this carefully. This might help you because this does not go in much detail. So if we are doing a regression model, okay. Next is you can simply do the pre-treatment in short. You can keep the cutoff here 0.99, and this is the data division. Which type of data division you want? You can select whichever mode you want to divide the data in. So, and then percentage of compound you want to keep in training set. You can vary also, you can keep it 75 or 80 or 65, whatever. And seed value, which compound you want this to place in test set. First one, second one, third one. And which method you want for the model development, MLR or data set. Okay, and then in the next step, simply assign the directory where you want the QSR model to be stored. Okay, and now you can generate several plots also, and these are some additional validation criteria, by scrambling method, and define applicability domain. It will calculate domain using standardized approach. And step 1, run. Within 1 or 2 minutes, the process what I have explained so far, which will finish within 1 or 2 minutes. So it has job completed now. The output. Now, see this. It has calculated the generated the step in the file. That's the problem. The input file what we have given, it was having some error actually. One problem was here in this software, one change is there. You have to place your activity column as the second column. Otherwise, this has taken this column as my activity column. So simply and we don't need this, and I will repeat that process more time, 1 more minute. This is a very useful tool wherein all the processes are clubbed in one file. And go to the next step, regression type. This is the treatment cutoff, division method, random or Canastone or Euclidean. I want to place 75% in the training set, and seed 1 should be placed. And GMLR. And the next step is you want the directory where you want to save the file. Okay. Is running. Job completed. Okay, now, then it is taken the same model. Okay, whatever the case, your response will come here and followed by your constant and coefficient and descriptors, and this will show you your R square value and Q square value. Here it is not proper because this has taken name as a response value column. So I had deleted that. I don't know why it came. Comma separated file. Here it is. Page. So the second column should be actually activity column. They're followed by descriptors.
So we have 5 more minutes for this session.
Yes. Okay, there is probably. Okay, at the end, the basic conclusion. And then all the validation metrics in one go. Additionally, it will give you your AD parameters in one go. You do not have to perform each step individually if you are using this software, DTC QSAR, in one go. As I have shown you in a graded manner, this entire process can be combined into one software. So with this, I finished the regression part, and I don't think there's a time for classification methods to explain. So you can see them. Yes. For the classification model, there's a manual provided. One can go through this manual. It gives you in brief how to proceed with and all, and it can help you to develop classification and regression model both. Okay. Thank you very much for your time, and now questions can be asked.
So the session is again open for the questions, if any. You can stop sharing the presentation.
So that switch it off there.
Beside that, one class sign is there. No, if you click on that, it will be stopped. So we will wait for 1 more minute if somebody want to ask any question or if somebody want to write in the chat box.
You can ask any question actually, not only related to final model development, or something related to QSAR or data preparation or data collection. If you need any other information related how we can generate data, from where we can generate data, collect data, you can ask.
So if there is no question, Let us thank Kavit and Professor Kunal Rai and his group member for a very nice hands-on demo. This demo is available on the YouTube. You can watch whenever you feel like. And whenever you have question, you can always write to Professor Rai and to Kabir also. Now the scientific session will end from this one. Only the hands-on participants presentation is left. That presentation will be taken from a separate link which is shared to only these 24 participants. So you can now sign out from here and you join it. All 24 participants, they are supposed to join from the link provided. Tomorrow morning we will start again at 9:30 a.m. where the talk will be there, and then one hands-on will be there in the afternoon session, and in the evening very lecture session will be there. So good evening and have a nice evening, and see you tomorrow morning. And all hands-on participants, see you in the new link one by one. Thank you.
Okay, thank you.